Hello, good morning, ladies and gentlemen. So, continuing the previous session's class. Yes, sir. This one?
Yeah.
Great. And writing is this? Okay. Great. Yes, sir. Sure, I'll do it.
You can use the right left.
Sir?
Right left.
Yeah, sure. No problem. Great. Hope everything is good, everything is positive, audible, visible. Please let me know if you have any issues, if any online people are watching this. Great. Yes. So, in the previous session we were talking about biotechnology. So, in that biotechnology we mentioned about traditional biotechnology, which largely is based on the process of fermentation, and this fermentation is brought about by certain fungi and also by certain bacteria, and through this organisms change their chemical composition, taste, odor, and these were used as products for human consumption, and this is referred as traditional biotechnology because of simple microorganismal activities. Next is the modern biotechnology, as we mentioned earlier, is completely based on deliberate manipulation or modification of genetic material, that is DNA or RNA or DNA and RNA, and synthesize new organisms or synthesize genetically modified organisms. So, please remember, if plants are modified, they are called as GM plants or GM crops, and if animals are modified or animal genetic material is modified, they are called as the transgenic animals. So, animals with transferred genetic material, transgenic animals, and in the case of plants they are called as simply GM plants or GM crops. So, we just use these terminologies to be specific, that is all, nothing else. And in the previous session we did learn about the working principle here, that is, we talk about certain enzymes that play a very important role in this process. Number one is the restriction endonuclease enzyme, which acts as gene scissor or molecular scissor, as we mentioned earlier, molecular scissor. This enzyme would cut the DNA at a specific site. Now, modification of this is found in some bacteria. This is an update here. Modification is found in certain bacteria. When bacteria are attacked by certain viruses, bacterial DNA will identify the virus genome, genes in a virus, and accordingly the repeating sequence of virus genome would be copied by the DNA, which is called as CRISPR. CRISPR means what here? Please understand, a bacterium when attacked by a virus, bacterial DNA would identify the virus genome and would prepare a fragment of DNA similar to that of this genome to next time whenever this virus attacks, this DNA will destroy it. That is called as CRISPR, which is clustered regularly interspaced short palindromic repeat or repeats. So what is a palindrome? Palindrome is which reads both ways. For example, none. In this direction or this direction, if you read, it reads the same way. For example, Malayalam. What is Malayalam here? M-A-L-A, Malayalam. So please understand this. Whether you read from this direction or this direction, it reads the same way. You will understand this. So this kind of sequence is called as palindrome. In English language we have this. In any language we will have this. Yes. Any word in Kannada? A palindromic word or term in Kannada? Vikatakavi is there one. But whether you will understand that or not. So any word in Kannada which is a palindromic word, which reads same way both in frontward or backward direction, or left to right or right to left. Think about it. You can message if you find one in Kannada. Yes. So this is a palindrome. Please understand this. Palindrome is repeating something both left to right or right to left in the same way. Now palindrome here in the case of DNA is what? The nitrogen base pair sequence. Please understand this very important concept here. In DNA structure we have deoxyribose sugar and the nitrogen base pair and then the phosphate group, and all these are called as nucleotide. You know this, yes or no? Polynucleotide is DNA. We know this. And if this nitrogen base pair sequence repeats, for example, AT, AT, TA, TA, if there is a sequence and if it reads in the same way, whether top to bottom or bottom to top, same way, palindromic sequence, in the same way it should read. If there is such sequence in the virus genome, that would be identified by the bacterial DNA and prepare a DNA which would be similar to that, and this is called as CRISPR. Clear? Now this CRISPR would identify with the help of a CRISPR associated protein 9. So this will be synthesized by the bacterial DNA, and next time this virus attacks the bacterium, this CRISPR with the help of CRISPR associated protein will destroy the virus by cutting the virus DNA or virus RNA at a specific site where there is palindromic sequence, and thus kill the virus. Did you get the point here? That's a concept which doesn't change. And we identified that bacteria are able to address virus attacks. Now we are using this CRISPR-Cas9 technology in the place of this enzyme. Now we are using CRISPR-Cas9 as gene scissor or molecular scissor. Did you get the point here? That is what is a recent development, that's all. And that becomes a UPSC question. What is CRISPR? What is CRISPR-Cas9? What is the use of, or what is the function of CRISPR-Cas9? And what are the applications of CRISPR-Cas9? That's it. All the questions have been asked. You get the concept here. Earlier what we used? An enzyme called as restriction endonuclease. For what purpose? to cut a DNA fragment. Why we are using CRISPR-Cas9 now? Because it is more specific, more precise, mistakes are less. Hence it is better, and you can prepare this in the lab at a low cost within short duration of time using bacteria. Hence it is quick, precise, no mistakes, and it can be used more advantageously. So that is why we are using CRISPR-Cas9 now, and no more restriction endonuclease. Clear? That is one aspect here. What is another enzyme we use in genetic engineering? Second enzyme we use is called as ligase. What is the function of ligase? Ligase fulfill the function of gene glue or molecular glue. That means to bring and attach two or more fragments of DNA together. That is a function of an enzyme called as ligase. So whenever you talk about modern biotechnology or genetic engineering or rDNA technology, we have certain raw materials, terminologies you have to understand, enzymes. Next, what you have to understand is a gene vector or DNA vector, which is nothing but a plasmid. What is plasmid here? Plasmid is circular extrachromosomal DNA fragment found in bacterial cytoplasm and plasmid. Next is what you have to understand is gene of interest. You find any gene with a specific desirable qualities in any organism, cut it, separate it, bring it, fuse with the plasmid, you can transfer this into any organism of your choice. Let me show you a simple picture here. We are going forwards or backwards, not able to understand. Okay, wherever you touch it goes that way, is it? Okay, great. A simple picture would help you understand this. Yeah, you can see that plasmid from bacterium, in which you have inserted the DNA gene fragment and transfer it to any cell. Here also you can understand this. So vector, that is plasmid, create a place in that by cutting with restriction endonuclease and separate the desired gene or gene of interest from any organism and join these two using ligase. We described about this in the previous session also, using ligase, and this is called as what? This is called as the rDNA, also called as what? Recombinant DNA. Yes, sir. Now, this rDNA is transferred into bacterium, and bacterium would multiply faster, so that you will get more copies of this recombinant DNA, and you can separate it and introduce it to any organism for your benefit. So this is a simple working principle of the genetic engineering or the rDNA technology. You can have a look at that. We mentioned about this earlier, if you remember. So plasmid is used and gene of interest is also separated, and both are combined, that becomes a recombinant DNA. This recombinant DNA is transferred into a bacterium so that you can multiply them into multiple copies. Bacteria multiply faster, and you can separate them and use them into plants to prepare GM crops or use it for any purpose, various applications, and the end effect depends on which DNA fragment you transferred through plasmid. That effect will be seen here. This is the working principle of recombinant DNA technology. That's a basic thing, that's a basic, please understand this. So, having that knowledge, now you can have a look at this picture. Now, we have had questions about this earlier. So, we had questions about this earlier. For example, red biotechnology, applications in health and vaccines. One more previous question I wanted to show you here. So, we had this question also. For example, on vaccines, we had a question earlier. Yes, difference between DNA vaccines and RNA vaccines. Previous question. Very important area. You can make note of all those things and prepare an answer should there be a question. Yes, just for your understanding purposes we have prepared this. Yes. Now this picture speaks a lot here. You can have a look at all these things here. Yes. So in all those areas for preliminary examination, such questions would appear. Match the following. Match. What is that? Red biotechnology, health. In a sense, medicines, vaccines and all. Yes. Green biotechnology, easy, anybody can answer this. What is that? Agricultural applications. White industrial, blue is marine, that is fish and seafood here. Yes. Yellow biotechnology talks about food related aspects here, and then grey biotechnology, environmental, brown for ecosystems in different regions, violet is ethical, legal or educational, and gold is for bioinformatics, and dark biotechnology is for like biowarfare, bioterrorism and all. So these are certain aspects what you can understand. Yes. So we have the same things here in general, just in the form of pictures so that you can remember them easily. So these kind of pictures would help you a lot, especially before going to the prelims. If you have a look at these pictures, you can easily remember these things and answer some questions. Yes. So as we mentioned earlier, what is that here? Traditional biotechnology talks about what? Fermentation. As we mentioned earlier, Carl Ericke or Carol Ericke, if you remember, we mentioned about that earlier also. Yes or no? He is the one who used the term biotechnology for the first time, traditional biotechnology. And then Paul Berg, who studied the DNA structure, and then he got Nobel in chemistry also in 1980. So Paul Berg is the father of genetic engineering or modern biotechnology here. So traditional biotechnology is based on fermentation, and modern biotechnology is based on recombinant DNA, also called as rDNA. Now you have some fair idea what is fermentation and what is rDNA, very specific. Yes. And then what kind of instruments do we use in rDNA technology? We use a restriction endonuclease enzyme. Just remember this: scissors, molecular scissor or gene scissor, just now we spoke of. Yes. What is the purpose of this? To cut DNA. To cut DNA. Can you see that here? Yes. And transfer that into a target organism with a ligase. That is one working principle of rDNA technology in general. Yes. And then various applications here, and applications would differ based on where we use, how we use. We will talk about them one by one. Yes. Hope this is visible to online people also. Yes. So this is working principle as we mentioned earlier. Just remember this picture, working principle. Yes, same thing. Yes, same concepts here. Yes. So next is the CRISPR concept here, CRISPR-Cas9. So it is also called as molecular scissors or gene scissors. So CRISPR concept is what? Whenever a virus attacks a bacterium, please understand this, very very important concept as we discussed earlier. When a virus attacks a bacterium, now bacterial DNA would synthesize this CRISPR location, which is palindromic sequence, and thus bacterial DNA identifies this and develops a CRISPR, clustered regularly interspaced palindromic repeat, short palindromic repeat, and accordingly this Cas9 would be developed, and we use that technology to cut a DNA fragment in any of the desired DNA. So we use this and we go for the Cas9 as gene scissor or molecular scissor, and that is how people studied about this and received a Nobel. They studied about CRISPR Cas9. So what is CRISPR? What is Cas9? CRISPR associated protein, and why it is used to cut fragments of DNA as the molecular scissors in the place of restriction enzymes earlier we used to use. So CRISPR is more precise, accurate and easy to produce in the lab from bacteria. So that is what you have to understand about CRISPR here in general. So we will come back to these vaccines at a later stage. Yes, next application of this CRISPR-Cas9 technology is what? The gene editing in general, gene editing, please understand this. So what do we do in gene editing in general? So what is gene editing and what do we do in gene editing? DNA or gene or chromosomal editing, which is the scientific principle behind something called as gene therapy. Gene therapy. Just understand this, that if a person, not necessarily a person, it could be a plant or it could be an animal also, but largely we use it for human beings. If a person or a child or an embryo or a fetus is identified as or diagnosed as having a genetic defect. We use lot of terminologies like chromosomal aberrations, genetic disorders, genetic defects. We use lot of terms here. Yes. So that means there is a defect in the structure of a gene or expression of a gene, because of which some unwanted proteins are there in the body of the person or necessary proteins are absent. If there is a defect at a gene level, it is difficult to treat unless you remove the defective gene or introduce a healthy gene into the body. Unless you do it, it is difficult to treat it. You have to understand this. So that is what we are looking at here. So that is a genetic defect here. That means it can be because of mutation. What is mutation here? Sudden structural change in DNA. It can be a point mutation, it can be a gross mutation. That means at a small gene or small nitrogen base there can be a defect or a change in structure, or gross mutation in large multiple areas there can be defects, or there can be chromosomal mutations also. Entire chromosome is defective. Mutations, or it could be because of what? Additions, deletions, in general, inversions. So we can have lot of such structural changes in the DNA of a person because of multiple reasons. It can be inherited from parents. Parents have defective DNA and you inherited it, or because of environmental factors like embryonic stages. As we mentioned earlier, if you remember, if pregnant women is exposed to X-rays during pregnancy, the embryo or fetus, unborn child, will develop genetic defects. We mentioned about this earlier, yes or no? Or if mother during pregnancy consumes alcohol, child will develop genetic disorders. So drugs, certain kind of drugs, sleeping pills also cause birth defects in children, genetic defects. So some reasons are known for genetic defects, sometimes we don't know the reasons. Yes. So in all cases, what's happening here? Unborn child is developing genetic defect. Now you want to treat, but you cannot treat it because genes are defective already. So externally you cannot treat it because you cannot cure it. Then only way to address that problem is through gene therapy. Then what is gene therapy? To treat some people with genetic defects because they cannot be cured otherwise. Then what is the method? What do you do in gene therapy? So it's a procedure. What happens here is, first we have to identify identification of the defect and the gene and its location in general, and identification of defective gene is done through markers, genetic markers here, and using CRISPR-Cas9 you cut that defective gene from the DNA of the person after identifying, and in that place introduce a healthy gene. In general. Now what did you do? You used principle of modern biotechnology, genetic engineering, to remove the defective gene and to introduce healthy gene into a person to treat a disease, to make that person healthy again. Now it is a treatment, hence it is called as therapy. Clear? Because you are treating genes, hence it is gene therapy. You will understand this. How do you do it? Using the CRISPR-Cas9 technology. And in this process, what are you doing? You are manipulating DNA. It is also called as editing. For example, you type something and some spelling mistake is there. What do? You will go and correct the spelling. You write an answer, you type an answer and you find some mistakes, you go back and correct the spellings or sentence formation or grammar and complete it. What did you do just now? What do we call this process as? Going back and correcting the mistakes of an answer or a typed material? Editing. It is called as editing, yes or no? For example, you are preparing question and answer, you are typing it. There is some mistake, afterwards you realized. You go back, identify the mistake, correct it and save it and complete the task. Don't you do it every day, almost regularly, whenever you are using computer? That is called as editing, yes or no? Similarly, here also it is called as editing. But what are you editing here? Genes in the DNA. You are correcting the mistakes. We don't know why that mistake has happened. Sometimes we don't know the reason. In medical science, there are two questions and sometimes we get answer to only one question. For example, there is a patient who comes to you, you are a doctor. Patient comes with a problem, health issue. Now patient will have two questions. First question is, why did I get it? Second question is, can you treat? Can you solve my problem? Two questions patient asks, yes or no? Imagine I am your patient, you are a doctor. I come with these two questions. Now what is important for both doctor and patient is answering only one question. Which question is that? Solution, treatment. Because prioritize, you have to prioritize whether why you got the disease is more important or whether we can solve it, treat it or not. If yes, how do we treat it and make that person normal? Whether that is more important. So which is more important? You can say both are important, yes, we know. But which is more important, which is an urgent matter? Which question should you answer immediately? Treatment, yes or no? But we do identify the reason also. We call it as a diagnostic medicine. Diagnosis means what? We have so many lab tests, blood tests, urine tests, and then we have radiological examination, X-ray, CT scan, MRI scan. We have lot of methods, yes or no? All those methods are to identify the reason for your health issue. But still, what precedes, what takes greater importance is treatment, yes or no? Likewise, genetic defects are present in many people. Now sometimes we know the reason, sometimes we don't know the reason. Sometimes a test, diagnostic test may be very expensive and may be time taking process. Till then, should the patient suffer or should you start the treatment immediately? Start the treatment immediately. That is what is. Gene therapy. Editing. Editing is what? You manipulate the DNA and remove the defective one and introduce a healthy one. You are not changing entire DNA of the person, you are not changing all genes of a person, you are changing only defective one. Hence it is editing, please understand. It is not replacing entire genome, it is only editing, small changes you are making just to make that person healthy, that is all, nothing else. That is the only intention. So that is what is gene editing here, and that is what you are looking at here. We are using CRISPR-Cas9 technology for gene editing. Now there is one more update or the recent development that is called as the base editing. So before we go to that, just have a look at it. Yes, gene editing. What are the different applications of gene editing? Should there be a question? For example, in the preliminary examination you get questions like what? Consider the following statements. They will give multiple statements. Gene editing can be used to produce sustainable biofuels, or gene editing can be used to produce hornless cattle. Whether cattle have horns or not, is it important? To some people it may be important, because horns get tangled in something, they fight and hurt each other, horns get problematic to some people. So hornless cattle is useful. So all statements are given, and the question comes, which of the above statements are incorrect or which of the above statements are correct? One only, 1, 2, 2, 3, 3, 4, or none of the above or all of the above. In that situation you should know all these applications, yes or no. That is why this picture, this slide is useful to you. If there is a question on gene editing and its applications, now you have some fair idea about what is gene editing. See, you do not have to become an expert to answer some questions in the UPSC. You are supposed to be a generalist, not specialist, yes or no. So you do not have to go into the microscopic details of gene editing, you understand this. But broadly speaking, what are the general applications? That is how questions come. Questions are very generic, please understand this. Yes, most of the questions are. So what is that here? Medical HIV treatment in general. So we can treat HIV using gene editing method. Okay, that is what you are supposed to know. A doctor would know how to do it, and in whom we cannot do it, in whom we can do it. You do not have to worry about it. Yes and no. Applications. Cancer therapy. On this we have had a previous question earlier, that is CAR T cell therapy. This was a previous question. Hence we would emphasize on this little more, little greater. What is that here? In general, CAR T cell therapy. Sorry. Yes, this was a previous question and recently news also. Hence we will learn little elaborately about this. Yes. What is CAR T cell therapy here? Yes. So what is CAR T, chimeric antigen receptor T cell? Okay, let me shed some light on it. So what is a T cell? Whether it is CAR T cell or not is secondary. What is primary is what is a T cell? T cell is nothing but T lymphocyte. It is one type of white blood corpuscle. It is one type in humans. We have two types of cells in the blood, isn't it? Of course, multiple types are there. RBC, red blood corpuscles, WBC and platelets we have. We know all these things, right? RBC are also called as erythrocytes. WBC are also called as leukocytes, and platelets are also called as thrombocytes. You know, basic, very basic stuff. Yes or no? Yes. So one type of WBC are lymphocytes. Lymphocytes are produced in yellow bone marrow. RBC are produced in a red bone marrow. So bone marrow is a soft gelatinous cluster of cells present in the middle of long bones. Very basic stuff. Yes. So lymphocytes are produced in bone marrow, but some lymphocytes mature outside bone marrow and some lymphocytes mature within bone marrow. So what? Accordingly, their nature, their function would differ. Those lymphocytes which are mature within bone marrow are called as B lymphocytes, bone marrow maturing lymphocytes. But some lymphocytes mature outside bone marrow. Where do they mature? They mature in thymus gland. What is this thymus gland and where is this present? Thymus gland is an endocrine gland which is present in the center, that is on the sternum bone. Sternum is like a tie, you know, here, from here to here it starts, you know, sternum, breast bone it is called as. This thymus gland will be active till puberty in humans. It produces a hormone called as thymosin, don't worry about it, which helps in the maturation of lymphocytes. Such lymphocytes which are produced by yellow bone marrow but mature in thymus gland are called as T lymphocytes. That is a T cell. In CAR T cell, what is T cell? Now you know. You understand this? What is the function of T cells or T lymphocytes? To produce antibodies. Please understand that these are basics, yeah. When would there be antibody? Whenever there is a, yes please, antigen. What is antigen? These are very basics. You cannot do mistakes in this. Yes, UPSC. Which exam are you writing? UPSC. Doctors write this exam. Engineers write this exam. Common people who have done BA, BSc, BCom also write this exam. Do you think this exam is same for all? It is same for all. Same question paper, same answer, same cutoff. I mean, for general, yes or no? And all people will go to mains. Mains also same for all, in a sense same papers, yes. After that interview, same for all, no bias. If you are a doctor, you will not get a different question paper. If you are an engineer, you will not get a different question paper. If you are a BA or BCom or BSc graduate, you will not get a different question paper. It is same for all. So it would be, some questions would be easy for some and some wouldn't be easy for some others. So others should study more, work harder. CSAT may be easy for maths undergraduates or engineers. Yes or no? It's quite obvious. But what about other subjects? Other subjects are easy for other graduates who are not maths undergraduates or engineers. So there is always some advantage to some people, some disadvantage to some people. So you have to work harder where you are weak or not very confident or familiar with, and it applies to all people. Are you able to understand this? So work where you are weak or not confident, other things you know. So some areas you should know about basics. If you are not from a life science background academically, not medical science, life sciences background, but still you should, that is why we say please go through NCERT books once. All these are present in NCERT books, school books. You understand this? First 1 to 2 months groundwork, homework. Moment you decide that you should write UPSC civil service exam, start with NCERTs. All subjects, 6th to 10th, you will have basics very clear. Make notes out of all those things if necessary in short form, so that revision would be quicker, faster. And current affairs you can write next to them, integrate them and solve previous year questions. You are ready for the prelims. It may sound simple, but it is not easy and same for all. It is different for different people, though question paper is same for all. Yes or no? So understand yourself first, where you are not very confident, where you are, strengths and weaknesses, you know, we say usually, what are your strengths, what are your weaknesses? Every person will have different things, yes or no? Weaknesses, if you identify, strengthen them. At the same time, maintain your strengths. And that is preparation we call. That is what you are supposed to do. That is what we have done to crack prelims. That is what every candidate will do to clear the prelims. It is not a rocket science or something. It is the same consistent work people do over a period of months or often years. You get it? But sooner you realize what to do, better for you, because in short time you would learn and crack it. You get the point, yeah? It's not something which is extraterrestrial stuff, it is pretty known stuff. But consistently every day you have to do it, and remember why you are doing whatever you are doing. These things are over a period of time, people change, and consistently you have to work, and not many people can work consistently over a period of time, doing same thing every day for years. Not many people do it. It's easy to start, but what matters is how you complete it. Finisher, not beginner, matters. You get the point. Anybody can start, yes or no? Anybody can start from anywhere. But who is the successful candidate? Who will reach interview, get into service, and come back and say how she or he has done it. Every person may have different methods to crack it. A person has cracked it doesn't mean that the same formula would work for you. There is no such rule. You are a different person. Your strengths and weaknesses are different. Know about yourself. Introspection is necessary sometimes, Look at your own self. Basics like a 1, 2, 3 numbers, numerical, and then alphabets A, B, C, D, E. Likewise, in science, these are basics, very basics. So, what is antigen? Antigen is foreign, alien, and potentially harmful. Potentially harmful. That's how your body assumes it as. And if something is potentially harmful, what would you do? You will try to protect yourself and try to neutralize the potential threat. One attempt by your body to protect its own self from potentially harmful things is by producing antibodies. And the ability of an organism to produce antibodies against antigens is called as immunity. What is immunity? You know that, yes or no? Immunology is the branch of medical science which deals with immunity. There are different types of immunity: acquired immunity, innate immunity, natural immunity, and then we have immunization, vaccination, lot of things. We will talk about them. Yes. So, of course, we have that also. If you want to have a look at that, see vaccines, DNA vaccines, RNA vaccines. Previous question, differences, and which are advantageous, which are not much, which are stable, which are not stable. Please remember this, from this slide alone we can prepare more than 10 questions, MCQs. So, have a look at, because some people will have photographic memory, just to help them these pictures, because if I speak you may not remember my words, but if you look at a picture you may remember the picture. In the exam what matters is what you remember, at least by looking at the question and answer. Question is given, answer is also given, yes or no, but looking at the right answer is not possible by all. It is there in front of you, don't you think? But still you can't see it. What's the problem? You think that you see everything because you have eyes. Try UPSC preliminary examination. Though it is in front of your eyes, you can't see it. That is why. So, at that time what helps you is cognition. Then in the exam you can recognize the answer. Second time. So, you are looking at T cell here. What is a T cell? Lymphocyte produced by the bone marrow, matures in thymus, hence a T lymphocyte, simply called as T cell. Okay, what is the function here? Immune function. What is immune function? Produce antibodies. Against whom? Antigen, harmful substance. So, immunity. Now you know this. If there is no antigen, would there be antibody? No need. You understand this? Okay, fine. So, T cells are important. T cells are important. T cells can identify many antigens based on their surface protein structure and synthesize suitable antibodies so that they can neutralize specific antigens. It is called as antigen specific antibodies. You will understand this. This goes a long way in immunology, in preparation of vaccines. For example, you take vaccine for a TB, BCG. Would that work for virus disease, virus infections, or fungal infections, or some other bacterial infections? Would that work? Specific. Based on what? Surface protein structure of antigen. That is called as specificity, antigen specific antibodies. One type of antibody works against only one type of antigen. There is no one size fits all approach. You will understand this. So immunity is very specific sometimes. Okay, fine. T cells, and T cells believed to possess immunological memory also. That is what is memory? Surface protein structure. You will understand this. So T cells play a very important role in treating or in resisting many infections in the body. Okay. Now what are we looking at here is CAR T cell therapy to treat cancers. You will understand this. Now with that basic idea you can understand this. What is that we are looking at here? Chimeric antigen receptor T cell. T cells, T lymphocytes are modified, genetically modified to recognize cancer cells and destroy them. That is what is the application. Please remember, it is a previous question. Important application of rDNA technology or genetic engineering. If you know it, it is better for you. So what is that here? First thing, what do? We take T cells from a person who needs a treatment. Please understand this. Yes. So we take from blood, we take T cells, patient or a person who needs a treatment. Okay. Next, what do we do here? Genetic engineering adds a chimeric antigen receptor to T cells. So a DNA fragment is added to T cells which can identify the antigen. What is antigen here? Cancer cell. That is called as chimeric antigen receptor. It is like a sensor you add to a machine, sensor to detect light or to detect distance, to detect movement. You add sensors. Likewise, you add chimeric antigen receptor DNA fragment to T cell you took from person. So that is deliberate modification, that is rDNA technology is being used here. Clear? Yes. Now we modify, so we modified T cells already, and these modified T cells are multiplied. Multiplied means we need more such modified T cells, so that we can introduce them again into the body of the person. They can circulate in the entire body and attack cancer cells. More is better in this case, because cancer usually affects many parts of the person's body. Then you need many T cells, hence multiply, and back to the patient supply these modified T cells which are CAR T cells. CAR means what? Chimeric antigen receptor, modified. Modified T cells are injected back into the person. So it is very much like your own body producing such specialized cells, so that your body will not reject them, because whatever foreign, your body would reject it, assuming that it is potentially harmful. It is like, how do you treat strangers who are standing in front of your door? You are at your home. There is somebody who is a stranger at your doorstep. Stranger. Immediately, how would you treat that person? With suspicion, yes and no. You suspect. You don't trust. Trust is not spontaneous. Doubt is spontaneous. Suspicion is spontaneous. You think that, hey, what if that fellow is bad fellow? You see, that's a human tendency. Of course, animals also. They get scared first, then they will trust you. Trust is not immediate. Trust is not automatic. Trust is not spontaneous. Trust is secondary. You understand this? Doubt is first. Suspicion is first. Fear is first. Hey, what if that fellow is a thief? What if that fellow is a criminal? What if that fellow is a bad person? You don't know anything about that person. Still you suspect. You have zero idea who that person is. Still you don't trust, but you doubt. Negatively you think about that person. Yes or no? That's an animal tendency. All animals do the same in the universe, known part of the universe. Humans are also animals first. Then whatever you think, you are next. Yes or no? So your body also responds the same way. Whatever enters your body thinks, hey, what if that is bad? What if? Well, let's prepare for the doomsday or worst case scenario. What if? And antibodies are produced. For example, you get sneeze. Sneeze is a simple example. You understand this? Mucous membranes, the histamine is released, swelling, water, all those things, sneeze, cough, what are they? Protective. You understand this? You inhale dust, your body tries to put it away through sneezing, assuming that it may be bad. Similarly, getting back here, CAR T cells which are modified, introduced back. Then what would CAR T cells do? Recognize and bind to cancer cells and destroy them. So this is in brief about stepwise process, how we use rDNA technology to modify your T cells so that your own T cells can kill cancer cells in your own body. It is one type of treatment. Treatment is also called as therapy. Therapy through what? Modified genetically engineered T lymphocytes, also called as CAR T therapy. CAR T cell therapy. Simple? Understood? Should there be a question, can you answer? That's it. Use this picture in the future. You get it? This was a previous question. An important application of rDNA technology in treatment of certain diseases, especially cancer. Okay, getting back here, we are again talking about same thing, applications. Applications, in that we spoke of what? Cancer therapy, how gene therapy helps in treatment of certain diseases. In that we discuss about cancer treatment. Is it clear? Yes. And then next is what? Sickle cell, of course. What is sickle cell here? We added that because that was also a previous question. Hence, we added that. Sorry. Yes. So sickle cell, we have that condition here. Let me talk about sickle cell here. This was also a previous question, hence we are elaborating on it. Sickle cell disease, also called as sickle cell anemia. Of course, both are not identical, but still related. So sickle cell. What is sickle cell? This talks about RBC. RBC is a red blood corpuscle. Don't say red blood cell. It is a corpuscle, formed element. Yes. So RBCs are also called as erythrocytes, are formed in the red bone marrow, usually are circular with a central depression like a discoid, peripheral bulged, central depression or concavity, or like vada, South Indian vada. Normal vada you eat in a hotel, central hole will be there, but in this hole will not be there, but depression will be there. Peripheral swelling, donut kind of, but not a hole in the center. Get it? Because they do not have a nucleus. Matured RBCs in humans are enucleated. Enucleated means without nucleus. Hence there is a compression, depression in the center. They are produced by red bone marrow, and after a time period of 110 to 130 days, average 120 days, they will go to spleen and die. Hence spleen, it will be here. Spleen is called as graveyard of RBC. Simple. So fixed lifespan, no nucleus, and they die. While they are alive, they will roam in the blood, bind with oxygen, transport oxygen from lungs to various parts of the body, to tissues, especially to mitochondria. Hence, they are also called as very important cells for your normal function. And RBCs contain a pigment modified protein called as what? A conjugate protein, hemoglobin. Together, heme is iron, globin is protein, conjugate protein. It is also called as what? Respiratory pigment, hemoglobin. You know all those things, yes. So, a normal RBC is a spherical discoid with central depression, spherical, and contains hemoglobin and a lifespan of 120 days and participates in transport of oxygen. Okay, fine, great. Now, what is sickle here? Sickle is a kind of instrument which is used to cut grass. Sickle. What do you call sickle in Kannada? Communist Party symbol. To cut grass, sickle. There is a handle. Hold this hand. You can cut many things with it, but usually people use it to cut grass. Yes or no? What do you call sickle in Kannada? Kutlu. Kutlu. Is it? Kutlu. So, this is kutlu. Now, we remove this. This is sickle shape. You can also call it as crescent shape or half moon, but depends. Yes. So, sickle shape. So, RBC because of defective formation process will have a different shape. Instead of spherical shape, the shape would be sickle, small, and does not have space to have equal quantity of this hemoglobin. So, spherical RBCs have good space for hemoglobin, healthy amount of hemoglobin, but sickle shaped RBCs do not have healthy amount of hemoglobin. So, low hemoglobin. Low hemoglobin means what? Low capacity to transport oxygen. That condition of low hemoglobin is called as anemia. Simple it is, you must have studied about this. Anemia. So, anemia means what? Suboptimal hemoglobin concentration in the RBC is called as anemia. Now, why there is a low hemoglobin here? Because of the sickle shape. Hence, it is called as sickle cell anemia. You understand this? And often this is an inherited defect. What is inherited defect? You got it from your parents or ancestors. Defective RBC production in the bone marrow. So, sickle shaped RBC is an inherited defect, genetic defect, and which can be treated using again gene therapy. Genetic defect, we can try, yes. And this sickle cell anemia, sickle cell disease is very common in people who are descendants of certain groups in the world, more common in certain groups, indigenous communities in many parts of the world, in Africa and also in India. Indigenous communities are also called as tribal populations. Tribals are like our ancestors, because we were also like tribals few years ago. We used to live in the forest, we used to perform hunting and food gathering, we lived in forest as groups, yes or no? We never had a technology, language or a script or phones and all earlier, no UPSC exam also, yes or no? Back then we lived a tribal life, but some people moved to a different lifestyle, but some remained as they were. You understand this? Those who remained as they were are still tribals. Other people who went away became non-tribals. So, whenever you come across any real tribal who lives in a forest, hunting, food gathering, simple primitive ways, please appreciate them, because it is not easy not to change. You understand this? They are consistent. They still are living the way we lived many years ago, and they are proud of it, and they say they are not wrong and they are happy. They are not poor, they are not backward, they are proud of whoever they are, whatever they do. That's not easy, you cannot do it. So whenever you come across tribals, appreciate them, try to help them out, understand them, don't disturb them. You understand this? Probably that's the best we could do, because they don't expect anything from you. You understand this? So tribals in general. Okay, anyways, so they are like our ancestors. You understand this? Okay, getting back. Problem is, many tribals in India suffer from malnutrition. Well, reasons are many, because of their culture or belief systems or unavailability of balanced diet, lack of awareness, whatever it is the reason, or changing forest biotic composition, animals, plants, hunting is prohibited, a lot of things. Yes, a lot of reasons are there. They suffer from, and somewhere some ancestor developed a mutation, sudden genetic change, which resulted in sickle cell RBC in them. And all this because the tribals follow endogamy. What is endogamy? They marry a person only within their group. They don't go for exogamy. What is exogamy? Marrying a person outside your social group, maybe outside your family, outside your community, outside your caste, outside your religion, outside your region, outside your geographical boundaries, outside your language, whatever it is. Exogamy. Yes? Exogamy is still considered as a taboo. Now what is a taboo? Taboo means what? What's a taboo? Forbidden thing. Forbidden. Forbidden or avoided. Hey, wrong, bad, sin. You understand this? So exogamy is still a taboo in India. But endogamy is practiced. Some people are racial also. Race, same skin color, same economic background, same educational background, same place, same language, same state, same caste, same subcaste, same religion, same region, same country at least. So people have their mindset tuned, conditioned to primitive thinking. And tribals, as we said, like we lived in the past, they still have that thought process that endogamy is right. What is endogamy? Marrying within their social group. A tribal person should marry another person who is also a tribal from the same group. They have their own reasons. Many people practice endogamy. Yes or no? Yes or no? Look at your parents. Well, most parents. Yes? Endogamous. Same community, same caste, same subcaste, same religion, same region, same language, same belief system, same traditions, same customs. Everything is same. Yes or no? Well, that's how it is. Endogamous. Endogamy means what? Same gene pool, population who have inherited same genes from same set of ancestors. Defective gene is present in all people. They marry each other, they have children, they also inherit same defective genes. Hence it runs in families. Hence they suffer from sickle cell disease, sickle cell anemia. Hence, if at all there is someone who needs immediate gene therapy treatment, is tribals in India. How can you say? Why you are saying that? Because look at the recent development. Birsa 101. It could be the next potential question. Birsa Munda, one of the greatest tribal leaders in India. Birsa Munda. So what happened here? So gene therapy treatment in India was developed, first ever CRISPR-Cas9 based gene therapy. To treat what? To treat sickle cell disease. You can ask a question, hey, why Birsa Munda name? Why not anybody else's name? There is nothing against anyone. But why that person? Natural it is, out of curiosity. You will get, why that person's name? Is there any specific reason? Yes, in India, across the world also, but in India more specifically, tribals suffer from sickle cell disease which is inherited because of their marriage practices. You get it? Because tribals suffer a lot more than non-tribals, tribal leaders' name was suggested. How appropriate it is? Yes or no? Hence a potential question. You will understand this. Birsa 101. So imagine this. So named in honor of Bhagwan Birsa Munda 150th birth anniversary in the month of November 2025. It is present in current affairs also. Current affairs also. First ever gene therapy also. Free to tribals in India. Free to tribals. Birsa 101. First ever CRISPR based gene therapy tool in India. Many reasons are there for it to be asked in the civil service preliminary exam this year. So that's how you can prepare. Yes. So that's a one time. It is one time therapy only. Only once you give, that's enough. That will treat the issue. A person need not to come to hospital frequently, because it is difficult for rural people, tribal people to visit hospitals every now and then. It is not easy, and they have a lot of challenges. Not like you stay in Bangalore and in the next street you have a hospital. Yes or no? But still home therapy, home services are still present in a city, because people cannot go. Imagine tribals from forest. No, we don't expect. Yes. So what is that here? So who developed it? CSIR, Council of Scientific and Industrial Research, along with Institute of Genomics and Integrative Biology, IGIB, and Ministry of Science and Technology. This is a potential question. Multiple statements will be given. Consider the following statements concerned with Birsa 101. It is a new tribal education policy. It is a new scheme for reservations for tribals in civil services. No, just mislead you. Birsa is tribal. Your mind goes in that way if you don't know. And they will say it is one CRISPR based gene therapy tool to address sickle cell disease among tribals, or prevalent among tribals, developed jointly by CSIR, IGIB and Ministry of Science and Technology. If you don't know, you will not choose that, yes or no. And then the question comes, which of the above statements are correct or incorrect? Incorrect is more dangerous question than correct, because you don't notice the incorrect thing. You will assume that, oh, correct. Very common mistake it is. If you have answered many mock tests or MCQs, you would understand this. Yes. So every box, everything is a potential statement based question. Definitely you will get this. Now you have some idea what is CRISPR-Cas9, yes or no? You have some idea about what is gene therapy also now. You have some idea about what is sickle cell disease. Now you have current affairs, right? Then only you will understand this, not without basics. So that's how concept, application and current affairs are integrated in a balanced manner. And hence, should there be a question, you can now answer it. Any question based on this picture, yes or no. So that's how it is. That's how it is done. Okay. Anyways, don't worry about it. Yes. So getting back here. So what is that? How we perform this gene therapy for sickle cell disease? How do we do it? Just for information purposes. Yeah. Yes. So we have bone marrow cells. We extract. Bone marrow cells means red bone marrow, which is concerned with production of RBC. That is the defect. There is a defect, because instead of spherical RBC, that bone marrow in that person, affected person, is producing sickle shaped RBC. You get the point here? Yes. You collect the bone marrow cells. You extract from large bore needle will be there. Extract it from this bone. You can easily do it. Infuse stem cells with normal gene for hemoglobin. So normal, that means stem cells which are preserved in the laboratory, which can produce normal RBC with normal hemoglobin. Take the DNA of that cell and inject it into sickle shaped RBC producing defective bone marrow cells. Bone marrow cells are there. Introduce this DNA which is concerned with a normal spherical RBC, normal hemoglobin producing capacity. That DNA is injected into the bone marrow cells. This is done in lab. Next is what? So, condition patient's bone marrow to receive new stem cells. Bone marrow cells are treated according to pH, temperature, and for some time they are kept, in the refrigeration. So conditioning happens, and once conditioning is there, the stem cell got attached to the DNA of bone marrow cells, then send them back into the person's bone marrow. Now the insertion of stem cells into patient bone marrow cells is complete, and that person's bone marrow now produces healthy spherical RBC with sufficient amount of hemoglobin. This is one method. Another method is we have bone marrow transplantation. Old method. What is bone marrow transplantation? Take all the bone marrow cells, take them out, every bone, major bones, long bones, femur, tibia, yes, radius ulna, femoris, all these long bones, surgical process, yeah, cut and remove all the bone marrow, give new bone marrow. That's expensive, laborious, and risk of infection is very high, and you have to visit hospital multiple times. But instead, CRISPR method, you know what is that here? It is only one time. So convenient it is. Yes or no? That is the advantage of BIRSA 101. Potential question. Now again, what we are looking at here is the applications of the CRISPR or applications of gene editing. We discussed about this, yes. So cancer, CAR T cell therapy is done, sickle cell, BIRSA 101 is done, yes, important things here. And of course, some basic things, drug discovery, research process, it helps in research processes, research. That means we can identify many conditions where gene editing can be used for advantage. We are researching about it, yes. So rice varieties which are drought tolerant. Drought means a scarcity of water, shortage of water. So drought resistant varieties of rice can be produced. That means you take the gene DNA which is present in already a rice variety which is growing in drought area. Take that DNA and introduce them into normal rice grain, and that rice grain becomes drought resistant. You can grow this in any place. You will understand this. So that is what is application. And then we have vitamin A rich banana, enriched. This is called as fortification, fortified foods, nutrition rich foods. Otherwise, banana doesn't contain much of vitamin A, but you can produce a banana with a high vitamin A content. Why you want to do it? You can prevent blindness among school children. 5% of the school children in India are prone to blindness because of deficiency of vitamin A. Deficiency. And just simply supply banana in the school which is enriched with the vitamin A content. Free. Yes. Every day you eat one banana with vitamin A, you can prevent blindness. Such easy and cost effective method. Simple method. You understand this. Application. In the entire country you can apply this, and you can prevent blindness in the future. You can save so many lives. Look at the potential of this application, unless implemented, of course. Yes. So hornless cattle. Hornless cattle. It may be less significant, but we can do it. Yes. So flu resistant chicken. You know that bird flu was a major issue a few years ago. Yes. Whether it is H1N1 or H5N1. We had a bird flu, swine flu and all, we had lot of issues. And not only birds and animals died, humans also got infected, and many humans also died. To prevent it, what? Flu resistant birds can be produced in general. Again, lot of applications. High ole groundnut. Oleum means oil. So groundnuts containing more oil can be produced using this gene editing. We can actually do anything. But we have ethical concerns. We can design our babies. A child can be born, we can edit the embryonic cells, and we can make the child having 200 plus IQ, or we can make the child to grow to a height of 10 feet. It is it is doable. For example, you must have heard about this. You must have seen the movie Jurassic World. Yes or no? Genetic engineering, they produce a dinosaur in the laboratory. That becomes notorious. Yes, Indomitable Rex. You must have seen this, Jurassic World. Genetically engineered dinosaur is Indomitable Rex. So which can see through infrared, which can detect through heat signature and smell, which can eat any organism, which can swim, which can jump, which has a lot of strength. Bullets cannot penetrate the skin, very thick skin. They modified it. Now that becomes a threat. You understand this? So kind of. So we can do many things, but again, why? If your why is good, how and what, we can work it out. Yes, spider silk production, please understand this. So steel stronger silk, silk produced by silkworms, which is strong as steel. Now not steel here, but very strong silk. Yes. So sustainable biofuels here, algae producing fuel lipids, algal fuels. We can make algae produce lipids here. They will be used as fuels in the future. And the targeted drug delivery systems, nano targeted drug delivery systems, small amounts of drugs are directly injected into affected area through algae, nano or microscopic algae here. And then better biofuel production, as we discussed earlier already here. Carbon pollution, that is renewable energy alternatives can be produced. Microbial fuel cells, using gene editing. And then the pollution monitoring tools. There are certain organisms which can indicate pollution levels. Again, algae only, genetically engineered algae. So please remember why this slide. Should there be a statement based question? Which of the following statements are correct or incorrect regarding gene editing applications? So this is almost comprehensive, barring one or two if there is a new development, current affairs. Yes. So this would be of great help to you in general, and we discussed about all these things in general. CAR T cell therapy also here. Yes. So this is about sickle cell disease and BRSA 101 also very important here. And a new drug has come into picture. New again current affairs, new thing. What is that here? Cas-JV or Cas-GEVI for sickle cell disease. Please understand this. Cas-JV, approved gene editing therapy for sickle cell disease that's intended to reduce the frequency and severity of pain crisis. Not yet into India. In India we have BRSA 101 only, but outside India we have Cas-JV or Cas-GEVI. It's again a gene therapy tool for treating sickle cell disease, but in India we have BRSA 101. Don't worry about it. So across the, it is a current affairs. You must have studied in current affairs, but still, just should there be a question? Cas-JV is associated with, and then answer options. It is a new space launch vehicle. It is a new drug to treat cold or flu, or it is a new solar powered, battery powered vehicle. You don't know, you have no idea. Cas-JV, you cannot guess. Or it is a new gene therapy or gene editing tool for gene therapy of sickle cell disease. Which one is right? Gene therapy tool, Cas-JV. You cannot guess it unless you know about it. That's gone. You get it? So that's how it is. So, or which of the following statements is correct about or regarding Cas-JV? Statement based again. That is not easy. You can go wrong. Yes. Just the term here. Yes, Cas-JV. Yes. So again, innovations you can have, again applications, same thing applications, you can understand all these things. Yes. So getting back here. Yes. Now this earlier it was a mains question, past mains question. What is that here? Ethical concerns regarding genetic engineering. Especially you can, as we mentioned earlier, what is that here? You can go for designer babies. Designer baby, that means you can edit and modify embryo. You can create new life, you can kill existing life, you can modify it. That means what are you trying to do? You are trying to play God, if there is any. Because people believe that all organisms were created by God, isn't it? The way they are. Biodiversity. Yes or no? Some people believe it. Yes. Now if you are creating life, modifying life and destroying life in the lab, you are also playing God. Is it ethically right? No. Hence it is a concern. But in China in 2018, a scientist misled a hospital to modify the embryos of humans, and they were born but with some defects, and then this person was caught as he performed experiments on human embryos without permission. He was sent to jail with fine also, but he proved one point, that it is doable. Yes or no? Nobody has done it before, he has done it and he became successful. Don't you think that is the other side of it? Whether it is wrong or right, or that is different. Nobody has done it, but he has done it and he became successful. Now that means that is something, an achievement, scientifically speaking. Yes or no? Whether you should appreciate it or you should punish that person, use that person's knowledge, expertise and skill constructively to treat some diseases which do not have treatment till now. Yes or no? Instead of putting that person in the jail, his expertise, skill and knowledge will be wasted if that fellow is put in the jail. Yes or no? Instead, put that fellow in the lab and give that fellow a challenge, that find treatment for this disease. Don't you think we can use expertise, skill in a more constructive and positive way, especially of that type of people? But simply China has put that fellow in the jail because that fellow tried to play God. Okay, but still, if you can believe an invisible God, why can't you believe visible and provable science? Yes or no? But again, people, media, they will write many things about it. Yes or no? Anyways, what is that here? Designer babies, eugenics. What is eugenics? A branch of genetics which deals with the selective breeding, selective mating of organisms to give birth to offsprings with desired or desirable qualities. For example, you see, eugenics can be simplified saying that a healthy person, a healthy male, healthy female are chosen so that their children will be born healthy. Is it wrong? No. A white skinned person married to white skinned person only so that their children will be born with white skin. Is it wrong? People practice this or not? Blue eyed person marrying blue eyed person and so that their children will be born with the blue eyes. People do it. A handsome person married a beautiful person so that their children will be attractive to look at. People do it or not? Film actors, they do. Yes or no? A rich person marrying another rich person so that their children will be born rich. People do it or not? You get the point? Selective breeding, selective mating based on desirable qualities. You get the point? IAS marrying IAS. Yes or no? Because somebody is IAS, you are marrying that person. Otherwise, no, you wouldn't marry. Yes or no? Selective mating. You get the point? Selective mating. If it is done based on, genotype. Genotype means genetic composition. And you select organisms based on specific genes and allow them to mate with another organism with a specific genetic composition so that their reproduction, their offspring will be born with a specific desirable genotype and phenotype. Then that type of experimental selective mating of organisms based on genotype and phenotype is called as eugenics, a branch of genetics, a branch of biotechnology also. Selective. People do it, but they have different names to it. Yes or no? You get the point here, context. If researchers do it in the lab, you call it as eugenics. But if you do it, you say it is your choice, decision for the betterment of future generations. Yes or no? So it's very common. If you do, it is right. If others do it, it is wrong to you. It's a universal phenomenon, isn't it? Yes. Others are always wrong in your story. Only you are right in your story. Okay. Because eugenics is highly criticized branch of biotechnology because of ethical concerns. How can you choose who will be born with what traits? Can you choose? Look at it, but with a different perspective. Who will mate with whom and who will be born with what characteristic features can be pre-decided. You understand this? That is a ethical issue. Who are you to choose the future of somebody? Every person should be free enough to choose his or her own future. Yes or no? But you are deciding everything about somebody who is not even born yet. That is a ethical dilemma. You get the point here. And certain genetically modified organisms may pose a threat to environment, may spread diseases which are not yet known to mankind, may kill native species, thus resulting in loss of biodiversity. You understand? We don't know. Hence there is a concern. We worry more about what we don't know than about what we know. Yes or no? Resistance. For example, in the recent past, we have had a current affairs issue called as antimicrobial resistance. You must have studied about this, yes or no? Many bacteria have developed a resistance towards drugs, medicines. They became multi-drug resistant strains, and now we have extensively drug resistant strains of bacteria which are also called as what? Yes please? Superbugs. You might have studied this in the newspaper. Current affairs. Yes. So, antimicrobial resistance is the resistance developed by certain bacteria against certain antibiotic drugs or antimicrobial drugs. Why? Because of irregular usage, irrational usage, unwanted usage and usage in undecided quantities or concentrations or dosages, and over the counter drug purchase and administration, self administration also, self without the prescription of a registered medical practitioner. Many people go and buy antibiotics and take as if they eat chocolates. Yes or no? This is the reason why some bacteria have developed resistance, and genetically modified organisms may be resistant to known drugs. What if they cause diseases in plants or animals or in humans? You don't have any known treatment. That means that's over for you. Resistance. And religious concerns. You know, religion is a very old social institution which brings many people together on certain grounds. Yes, and rules, traditions, customs, beliefs. Yes. If anything is new, if anything is not in consonance with religious scriptures, that may be criticized by the religion. Yes or no? As blasphemy. If you are creating life in the lab, you are opposing God. Yes or no? You are not only opposing God, you are criticizing God, you are condemning God. That is anti-religious. It is called as blasphemy. You know what is blasphemy? Religious offense which is punishable. Blasphemy. That's a general English word. Yes. So, religious concerns. And next is what? Definitely caste. Why should expensive things be of ethical concern? Please remember, this can be a GS4 question also, your ethics question also. Is expensive thing an ethical concern? If some people cannot afford some things, is it ethically wrong? The price of some products of being very high, is it wrong to have very expensive products in the market? Yes, it is. If it is about essential things, water, food, shelter, clothing, these are basics. If these become expensive, then they cannot be affordable and accessible to many people. So many people would experience discomfort, loss. Now that is ethically wrong. Only few can buy, that is different. If it is a luxury thing, only some people would want it or need it. Need is secondary, want it. Yes or no? For example, health care. If it is too expensive, don't you think it is ethically wrong? Health care is mandatorily available, accessible, affordable to all. But only few can afford now some treatments. Don't you think it is ethically wrong? In that sense, it is of ethical. Please remember, it is a mains question. You have to elaborate it of 150 or 250 words. So use your creative attempts and then expand it. Okay, anyways, don't worry about it. This could also be a preliminary question also. Which of the following statements are correct or true or incorrect about ethical issues related to recombinant DNA technology? They will give statements. Now that could be a preliminary question. Yes or no? Of course, yes. So this is about your some recent developments and concepts and applications of recombinant DNA technology. Clear? And some potential questions. You understood. Next, we will move on to next thing. What is that here? Second, application based. Nanotechnology is emerging very fast, and in almost all fields, let me show you some picture. In almost all fields, there is application of nanotechnology. In almost all fields, you can see this here. Like earlier, we have shown the picture where applications of biotechnology, recombinant DNA technology are there, from which you can get a question. Same slide, similar slide is this, applications of nanotechnology in various fields, and from this you will get a question. Statement based questions are the trending UPSC preliminary questions. Match the following statement based, which of the statements are incorrect, which of the above statements are correct. That is what is trending. Yes or no? Now, you can create more than 10 questions, statement based questions from this slide alone. So that is why more content is given for you in the form of a single slide. So it will be easy for you to revise just before the exam. Yes or no? So that is the purpose of our presentation here. Yes, IT, information technology. We live in Bangalore, the IT capital of India, Silicon Valley of India. One more is getting ready, Mangaluru, Silicon Beach of India, it seems. You know Mangaluru, it is a next IT capital hub for India, apart from Bangalore. So that there will be less of stress or pressure on Bangalore. So people in Bangalore can live with a little amount of peace, that's just to take the pressure off. Some went to Hyderabad already, and some are going to Mangaluru, some are going to Chennai. So Bangalore can breathe some air and move fast in the traffic, Anyways, getting back here, various areas where we can apply nanotechnology. One previous question, because I found it very common and regular thing. Sorry. Okay. It's not working? Great. Not working? Excellent. Is it remotely controlled? Okay, yes. Okay. So, nanotechnology, we had one thing here. Just wanted to. This is a previous question. Targeted drug delivery system, targeted drug, nanorobotics. Previous question. Camptothecin also, camptothecin previous question. Cancer therapy, camptothecin, anti-cancer drug, previous question. One thing was there which I wanted to show some important thing. Wearable electronics, this is also previous question. Wearable electronics, you can wear your phone around your wrist. Phone, it becomes graphene screen, it becomes flexible. Wearable electronics is a previous question here. Yeah, your phone has a tempered glass. Yes or no? That has nano coating. It's a previous question. SSC, I think, staff selection commission question. Yes, nano coating of Teflon, this tempered glass. Yes. Now that was based on what? It's a Corning Gorilla Glass. Yes. These layers were asked as a question. Can you imagine that? These layers. You get it? These layers. Nano coating. This was a question. So I just added all the previous things in the form of slides, So these are, and then of course we have a lot of applications here. Yes. So nanotechnology. What do you mean by nano? 10 to the power of -9 size. Just for your understanding here. Yes. Nanotechnology. Yes. So let's have a look at them. Some information for you, so that you can answer some questions. Yes. So what is the concept here? Nanoscale materials. So ranging from 1 nanometer to 100 nanometers. So dealing with materials and their properties of the size of 1 nanometer to 100 nanometers. That is a nanotechnology, using them for various applications. Yes. So we will try to move on here. We will try to show you how the size is for comparative purposes. For example, a coin 1 millimeter, RBC 10 micrometers, virus 100 nanometers, and Buckminster fullerene, very very important, which is the building block of carbon nanotubes, is 1 nanometer in size. Now that is important for you, please understand. Just comparison purposes here, for the purpose of imagining what is nano for you. And a very important slide will be this here. What is that here? So nano silver, titanium dioxide, anti-wrinkle, anti-aging here, fullerene previous question, carbon nanotube previous question. One slide can give you answers to 2 to 3 questions. You get it? So that is how we have. So nanotechnology, how it works in general, very basic thing here. So you can understand this. Yes, 1 nanometer is 10 to the power of -9 meters, a billionth part of a meter. Meter as one measure of length. So a billionth part of this 1 meter is 1 nanometer in general. So what will happen because of reducing size in general? What will happen here? The concept behind nanotechnology, why it is important, how nanotechnology is advantageous, on what basis, what is the concept behind it. So 2 things here. Number 1, when matter becomes smaller, its physical, chemical, that is molecular properties change. They change, please understand this. This is applicable to all matter, all objects, all materials. If you reduce the size, physical and chemical, for example, you see, you take any piece of metal, if it is thick, you cannot bend it. If it is thin, you can bend it. So look at this, physical and chemical properties. Are you able to understand this? Yes. Likewise, for example, if gold is converted into nano material, nano gold can dissolve in water. Are you able to understand this? Now you can ask me, hey, why you want to dissolve gold in the water? That is different. Now what we want to prove is, by changing the size of matter into nano, you can change physical and chemical properties of the matter. To prove that point, we are mentioning this, that's all. Are you able to understand this? For example, why 90%, more than 90% of the diseases human beings get, they get from microorganisms? Because of small size of microorganisms. For example, lion or tiger can kill many humans, yes or no? But you know who is killing many humans? Microorganisms. Why? Because they are small. You understand this in general? Okay. For example, COVID-19 must have taught people many lessons. The future enemy need not be big or making loud noise or explosive or visible. The enemy can be so small and invisible and can silently enter your body and kill you from inside. That will be future of warfare, biological warfare, bioweapons, bioterrorism. Do you understand this? That is actually smart way, easy way. A lab of this size, you modify some microorganisms, genetically engineer them, make them toxic and release them into the water or air. Overnight that would infect many people, and within few days many people will die silently. Nobody knows. That will be the future of weapons and warfare. You understand this? There is no escape from this. Don't worry about it. How? No, that's over for you. The only thing is, see, engineers have become terrorists, MBAs have become terrorists, yes, uneducated illiterates have become terrorists. No doctor has become terrorist till now. If a doctor becomes terrorist, that's extinction level event to humankind. No human can survive a doctor's brain. Doctors not only can save lives, opposite and also can be done, and doctors know better. Yes or no? Osama bin Laden was engineer and MBA. You know that? His methods were little technical. But no doctor has become a terrorist till now. Yes or no? But what if? What if? A future. In the future. We never know. That's how it is. Anyways, don't worry about it. So when matter becomes smaller and smaller, their physical, chemical, molecular properties change. First point. Smaller it is, different it would be than its larger version. That should be simple enough for you. Yes. Second point. Smaller it is, smaller it becomes. That means in terms of size. More surface area it will have. Please understand this. For example, if an object is made into smaller components, if an object is converted into smaller components, it will have more surface area. So what will happen if there is more surface area? It can occupy less space but can have more influence. That means it can absorb more material, it can cover larger areas, less quantity but cover larger area. Are you understand this? And this is also called as quantum size effect. For example, coatings on surfaces. Usually paint, paint we coat on surfaces. Yes or no? It's very simple and direct. You coat with a brush. Depending on the size of the bristles of the brush, amount of paint which can be applied varies. Yes, and the thickness of paint coat also varies. If you use nano paint and use a machine to spray nano coating of the paint, the amount of paint, quantity of paint consumed would be much lesser, but can cover equal amount of surface. So with less quantity you can cover more surface. You understand this? That is one. For example, say you have one silver plate, silver, yes, argentum. You want to cover it with gold covering. People do it. So you need certain grams of gold. You have to melt it. Goldsmith will do it. Melt certain quantity of gold, yes, and take a brush like structure and apply the coating on the silver plate, certain quantity. But if you have a machine which can use 1/100th of the quantity of gold, convert it into nano particle size and spray it, very less amount of gold can be used to cover the surface of the silver plate. Yes or no? So that is what is the meaning of what? larger surface area. This applies to medicines also. For example, you take medicines of some milligrams, yes or no, or some grams also, big tablet you take. Some of that tablet will be wasted, it will not be absorbed. Some of it goes to kidney, some of it goes to liver, some of it goes to the target area. Yes, some of it goes as undigested waste. Carrier particles will be there. Yes, so large quantity, big tablets. Sometimes some people find it difficult to swallow. Old people, it will be stuck in the neck, throat. Yes or no? Okay. Instead, if you can convert that into same quantity of same medicine into nano size and directly deliver it to affected part, you can save a lot of other things. Yes or no? That is the surface area of small size advantage. Yes. Okay. Apart from that, quantum size effect is the third advantage of nanotechnology. What is quantum size effect? When objects are converted into nano size, they develop a unique property called as molecular self-assembling units. That means nano-sized particles, if you arrange them, they will assemble, they will arrange next to each other, they will arrange themselves in such a way that they will become attached to each other by themselves. Molecular self-assembling units is one unique property observed under quantum size effect. Nanoparticles, nano-sized materials have the capability to self-assemble. That is a unique property observed by various scientists when matter was converted into nano size. Self-assembling molecular. This why this is useful? Because there is an application called as, similar to MEMS, there is a new application called as NEMS now. Earlier MEMS is there. You must have studied what is MEMS? Micro electromechanical systems. Small size machines. Yes or no? Micro electromechanical systems. Now, because of this property identified, now we have NEMS, nano electromechanical systems, which is similar to, as we have shown the picture for you, nano electromechanical systems. What you are looking at here is RBC. Please understand this, RBC. This is a NEMS. What is NEMS? Nano electromechanical system, also called as nano robot. NEMS. A simple example of NEMS. This can deliver drug like iron. For example, if a person is suffering from iron deficiency anemia. Many people suffer from it. Yes. So, we prescribe them iron tablets or iron syrup, which gives side effect, headache. You understand this? How to avoid it? You cannot avoid it actually. You have to give one more tablet for headache and your entire life will be filled with tablets. That's a routine. Many people would experience. Yes, na? How to avoid it? Can we take the help of nanotechnology? Yes. How? NEMS can be infused with a nano iron. How much ever each RBC requires, only that much iron would be supplied directly to RBC. No side effect, zero side effect. NEMS, targeted drug delivery systems, can be developed using nano electromechanical systems, which is based on quantum size effect, molecular self-assembling units. This is just a glimpse of just one application. We can have many such applications. Cancer drug can be delivered directly to cell. For example, we mentioned about it, camptothecin. We mentioned about camptothecin. We have that picture also here. Camptothecin is a cancer drug. Camptothecin is a cancer drug. Camptothecin, anti-cancer drug, and this can be delivered directly to cell through mesoporous silica nanoparticles. Why we need to use this nanotechnology directly? Why can't you inject camptothecin? Cancer chemotherapy has many side effects. Hair loss is one. You see, cancer people lose hair. Before they lose hair, they shave off their head, because it would be embarrassing. Yes or no? You cannot accept your hair, you losing your hair. Hence, many people shave off their hair. Yes or no? You must have seen. Cancer means no hair on the head. You get the point here? Yes. So, side effects are many to cancer chemotherapy. How to avoid them? There is no negotiation in cancer chemotherapy. You cannot say no to drug, because otherwise you will die. There is no way out. But now with the help of nanotechnology, we can directly deliver anti-cancer drugs to cancer cells. No side effect. A person will be absolutely normal and cancer cell dies. Can you see all those things here? Cancer cell dies, you are healthy. Simple, no side effect, nothing, nobody would know, and you wouldn't even experience anything, any discomfort. Safe, easy. That's completely a game changer, isn't it? If applied appropriately. You understand this? That's the idea here. Anyways, getting back here, what is that we are looking at here? That is one application of quantum size effect. That is just one application. There can be many such applications. Yes, quantum size effect is when you convert matter into nano sizes, they develop the ability of self-assembling. Molecular self-assembling unit is a property of nano-sized materials which can be used to develop NEMS. NEMS is nano electromechanical systems. Nano electromechanical systems, also called as nano robotics, a new branch of robotics. These robots can enter places where no other equipment or a machine or a human or any organism can enter, and do whatever you want them to do, remotely controlled. Remote, you can control them remotely. You can get the job done and nobody knows about it. They are fire resistant, you can send them into the fire. They are radiation resistant, they are darkness resistant, they are water resistant. They can enter your body also, enter any part of your body, do the job and come out without harming your body. That's miraculous. We can send them into space also, where no human can survive, and study everything and come back. So applications are infinite. You understand this? But idea is important. That is a concept, because concept doesn't change. Applications might change, concept doesn't. Please focus on it. Yes. Now, having said that, let's have a look at what is nanotechnology. Of course, we have had previous questions earlier. Would you like to take a break now before we start nanotechnology? Say yes or no. Break? Okay, take a break for 10 minutes. We'll try to come back. For online people, yes, thank you. See you after the break. can talk about nanotechnology. Yes, we are talking about nanotechnology here. So, the principles of nanotechnology, the scientific principles, as we mentioned earlier, that at nano sizes, properties, as we mentioned earlier, the physical properties, the chemical properties, and of course to some extent, if there is any, the biological properties of materials would change. Now, how do we explain this better? That means to understand the applications better in general. So, this is based on certain scientific aspects. Number one is, as we mentioned earlier, the quantum size effect, small size effect or nano size effect, quantum size effect. First one is a very simple mechanical. What is that here? Higher surface area to volume ratio. Now, what will happen because of this is, though small quantity of the material is used, its ability to bear strength increases. So, small quantity becomes stronger, which is actually opposite to what the normal modern physics principles would prove. Because you see, to make something stronger, we make it bigger, thicker and wider, yes or no? Whether beams or pillars or girdles or whatever it is, yes or no? Thicker, bigger is considered as stronger. That is a normal physics principle, isn't it? More particles can bear weight, yes. But here is, once you increase surface area when compared with volume, more particles spreading in larger area can exhibit greater strength, which is opposite to the classical physics principles here. Strength increases. That means, that is why you see we mentioned about it earlier, that if you want to observe that carbon nanotubes in general, whenever you have this carbon nanotubes, yes, they are very strong, they are nanomaterials and fullerenes also. So, they are very strong, though they are nano sizes, they are so strong that tear resistant textiles, stronger and lighter sports equipment can be made. They don't break. So, they have this property, as we are talking about it, they have this property. Higher surface area to volume ratio increases its strength. That is one aspect here. Second aspect is its energy conductivity. Please understand this. Its energy conductivity. One simple formula, maybe you would have studied this, I want to mention here is, there is a concept in geography called as, you must have studied red shift and blue shift. In geography you must have studied this, it's a old concept. In geography it's a pretty old concept, it is not something new. What is red shift here? When object moves away from you, it appears red. When an object comes near to you, it appears blue. Simple. Yes. It is based on the physics principle, hence we are talking. It is physics is science, hence we are talking about it. It is based on Doppler effect. Doppler effect. When object moves away, any object, usually in geography and astronomy we take celestial objects. Celestial means cosmic, in the space. When any star or any cosmic entity or a meteor or a satellite or a planet, whatever it is, if it is moving away, it gives a reddish appearance. Far things are red in color. If an object, cosmic celestial object is coming closer, it appears little bluish. Well, how it is related to science, you can ask me a question. Yes. So when object moves away from what? Away from the observer. Away from the observer. It could be a camera or a satellite or your own eyes. Get it? So moves away, it appears red. And when an object moves closer, it appears blue from the observer. So Doppler effect. It is similar to sound also. Sound also follows a Doppler wave. Yes, formula. Intensity of sound increases when the source of sound approaches the listener, and intensity of sound decreases when the source of sound moves away from the listener. It is simple Doppler effect. Based on which you have ultrasound scanning, Doppler scanning occurs. It works. Now here, how it is related to science here is electromagnetic spectrum. We studied about this earlier, that amongst visible spectrum in general, you have infrared and ultraviolet. Yes, the wavelength decreases as you go from left to right in electromagnetic spectrum, whereas the intensity frequency increases when you go from left to right. Please understand this. We mentioned about this earlier, yes or no? So this microwaves, radio waves, infrared, invisible, non-ionizing, this visible spectrum, and then we have invisible ionizing. So wavelength is higher on the left side. Radio waves, microwaves, infrared have greater, longer wavelengths. Longer, that means they can travel longer distances. Whereas on the right side, ionizing radiations, their wavelengths are lesser, shorter, we say, short wave. So when we say, when object moves away, it appears red, means higher, longer wavelengths. Long wavelength energy they release. When objects are moving away, they release long wavelength energy. When objects move closer to the observer, they release short wavelength energy. Short wavelength. For example, blue is having shorter wavelength than red. Red has longer wavelength. Hence, farther objects, long wavelength, can travel longer distances, appear reddish, called as a red shift. And closer objects, because of shorter wavelength of energy released, appear bluish, blue shift in geography. What is the significance of this in geography? When you observe stars or celestial objects, they appear red. What does that mean? That means they are moving away from Earth, or Earth is moving away from them. So what? That means universe is expanding. That is the inference we can draw. Universe is still expanding. Hence objects which are farther are appearing red now to people on the Earth. You will understand this. That can also explain why sun is red. You understand this? In general. Okay. So that is not necessary now here. But again, how it can be applied? For example, gold. What is the color of gold? Oh, come on. Do you have any doubt in that? Why gold is precious? Because it is yellow. For example, you paint gold. You take 1 kilo, or 1 kilo is too much. 10, 15, 20 or 100 grams gold. Take a gold biscuit or bar, color it black, throw it on the road, but observe, because you know that it is gold, nobody else knows. Color it black, throw it on the road, not even a beggar would pick it up. I have waste. Take any object which is not gold, take iron, color it yellow, throw it on the road, everybody would pick it up. A simple experiment. What does that prove? People go by looks. People choose something purely based on looks first, then only whatever it is next. That is one point you can prove. Second point is yellow. Gold is precious, gold is valuable largely because it is yellow. You understand this? For example, platinum is actually more precious than gold, but people don't like it much, people don't buy it much, people don't save it much, people don't wear it much. Why? Actually, platinum is more expensive, rarer, more precious than gold. We know that. But not many people are crazy about platinum as they are crazy about gold. Gold is cheaper when compared to platinum. If you want to be really rich, if you want to look rich, if you want to feel great, then you should buy platinum. If you really think you have money, then why people are not doing it? Platinum color is not as people think as attractive as that of gold, because yellow, again it will go on yellow. But when you convert gold into nano gold, gold appears red. Now that is something. Bulk gold is yellow, nano gold is red. Change of property. That means nano, what does this infer? What is inference? What do you understand based on this? A very important point here, because this can be a potential question. I don't know whether UPSC will go to that depth or not, but you never know. Yes or no? You never know. This proves that the nano size particles, quantum size effect we say, again quantum size effect. Okay, let me ask you one more question. One more question based on your whatever knowledge you think you have. Which among these represents in general more amount of energy? Yes, please. It shifts. We know that energy shifts. Yes or no? Usually when you heat objects warmer or hotter, they become, change in color happens. For example, put iron in the oven or iron in the furnace, it turns red once it becomes hot. So red indicates a higher energy state. Yes or no? Usually, yes. So red hot we say usually, something as red hot. We don't say violet hot, indigo hot, blue hot. We don't say that. Yes or no? Red hot. That means it emits more energy. Danger is denoted with red. Stop signal is red. Danger is red. Something bad is red. Yes, hot is red. So we denote high energy things with red. Yes or no? There is a meaning, there is application to it. What does that mean here? That quantum size effect also says the same thing, that when material is converted into nano size, their energy which can be transferred, energy transferring ability also changes. For example, when particles are closer to each other in bulk products, in macroscopic large objects, when particles are closer to each other, when they are large, the energy bandwidth available between them is shorter. In simple terms, try to understand. Energy bandwidth, for example, there is energy here. This has to absorb the energy and transfer the energy, and the distance is so less because it is a denser macro large. Because particles are next to each other, energy bandwidth between each atom, each molecule is shorter, hence energy bandwidth is shorter smaller. So amount of energy possessed and transferred by each atom is lesser. You will understand this. Okay. Convert this into nano. You understand this? Now it becomes less denser. Atoms move away. Higher surface area. Yes, move away. Yes, thin. You know that. Small. What happens between them is each particle will start generating more amount of energy because energy bandwidth became larger, wider, greater. Hence we say nanoparticles, because of quantum size effect, would conduct more energy and can transfer more energy than bulk particles. Okay, so what do you want to prove by saying this point? Saying that nanoparticles can emit more amount of energy than their bulk counterparts because of higher energy bandwidth available because of quantum size effect in nanoparticles. This indicates the strength of nano-sized materials. They will have greater strength and they will conduct more energy. More energy means efficient energy transfer without any loss of energy. So what? That means electrical conductivity. Electrical conductivity is greater. Now we can use this technology in semiconductors, transistors and microprocessors to conduct energy. So more efficient electrical conducting systems which are small size can be built. That is the application. So computers and other electrical equipment can be made into small but more energy efficient. Can we produce such systems? That will change market dynamics a lot. Yes or no? That is the application based on the principle. And one more. For example, nanoparticles. Quantum size effect again. Nanoparticles. Depending on the type of nanoparticle you are using, the color shift and the amount of energy released by them because of supply of electrical energy also would be different. Hence these are used as multiple layers in displays. So colors would become more vibrant and thinner. So thinner, more energy efficient and vibrant displays can be made. Displays of electronic devices. For example, watch, phone, television or any screen can be made thinner, more energy efficient. That means it doesn't become hot or warm, and more colors it can transmit, more vibrant. So if it is very thin, it can be flexible also. Wearable electronics and thin LED, QLED TVs, quantum LED. Heard about this? QLED. Nowadays QLED TVs are coming, very thin, like a wall painting, very small wall painting kind of, very thin. What kind of displays are used? Quantum. Quantum means what again? Nano. Are you able to understand this? That's application. That is application. But underlying principle is quantum size effect. Please understand this. Quantum size effect. So it can transmit energy because of nano size. And usually nanoparticles, based on their altered property when compared with their bulk counterparts, can have either red shift or blue shift. Either red shift or blue shift, or would move between red shift and blue shift. You will understand this. Hence, they change colors and look vibrant and thin, energy efficient and vibrant. Now, this is a desirable quality for displays. High quality display, low energy consuming display, thin display and a vibrant display. That means it is a good marketing tool to increase the price, decrease the size, increase the quality. QLED displays are costly, but they are very thin, very colorful and valuable. Look at that. You just changed something which completely changed market dynamics. Okay, quantum size effect. And also, as you can see that here, energy transmitted by quantum dot sized material also is greater. Hence, they are thinner but stronger, flexible yet stronger. You can imagine, it is very advantageous. Thin and strong, flexible and strong, that is very advantageous feature. You can apply this to many places. For example, nano silicon coating on structures. Please understand this. On cement structures, nano silicon coating would increase in general. So, of structures, cement structures, walls, all those things, nano silicon coating will make the wall waterproof. Nano silicon coating, nano coating would make it waterproof and also very attractive but strong, and the paint and the structure will not get damaged because of extreme climate or weather events. So, in structural engineering, you can see that here silicon dioxide, you can see that mechanical strength is increased, as we mentioned earlier. Yes or no? So, nanoparticles, because of quantum size effect, will have more strength but thin size. See, silicon oxide, nano silicon, mechanical strength is increased. Now, in the future, such big buildings, pillars may be smaller than what they are now, using nanomaterials, because they are strong but they occupy less space. Wall between rooms can also be very thin but very strong, using nanomaterials. So, in the future, you will have more space but stronger structures using nanomaterials. Are you able to understand? Applications are infinite, you can say that here. Yes. So, that is what. And can we build earthquake resistant structures using nanomaterials? Because they are strong but flexible. Now, applications are infinite. Can we build structures on the water? Can we build structures in the earthquake prone regions using nanomaterials? Now, you can change the future. You will understand. Applications are infinite. If you know working principle, you can understand applications very easily. So, that is what is one idea here in general. So, that is what is a working principle, that is the technology involved behind nanotechnology. Please understand this working principle. So, if you understand that, you can understand everything else very easily. For example, all these can be understood because you understood the principle. Now, you can understand what is that here. Very simple. Smaller, faster, more energy efficient. Now, you know. You can easily correlate, yes or no, and you can easily answer it. Smaller computers, efficient computers, less energy consuming computers. Possible? Yes, of course, possible. And you know how. Yes. Next is medicines. We mentioned about this here. You can deliver nano electromechanical systems, can deliver drugs to the specific area without wastage. No side effects. Possible? Possible. You know how. And then consumer goods, for advanced packaging material, sensors and lab on chips for food quality testing. Very thin sensors can be arranged on food particles. Yes, possible. And then energy, more efficient, cost effective technology for energy production. Can we produce them? Yes, we can, of course. Now you know how. That's it. Entire slide is clear to you. Simpler than it was before. That's a simplification method in general. Yes. So that is a quantum size effect here, and then yes, we can have a lot of applications as we mentioned earlier. A lot of applications. You can understand each one better. Yes. And this for comparison sizes here. Okay. So we are done with all these things. Applications, you can, yes, applications in various areas, and few of the previous questions I wanted to mention here. Nano silver, sorry, yes, nano silver. So we know that silver is a disinfectant material, it kills bacteria and fungi and all. That is why silver coating is preferred in water filters and all. Nano silver coating, yes, possible. So fungi, algae and bacteria can be killed. So these particles can accumulate, that is the impact here. So used in water purifiers, deodorants and washing machines. Now we apply the same thing, we apply the same principle to the nano urea, yes, this one, and neem coated urea. Both are previous preliminary questions, UPSC Civil Service preliminary examination. You understand this? Nano urea, it is to save the urea, because urea is a source of what? Yes please, source of nitrogen. It is a very popular fertilizer in India, and often the price of urea determines the profit of agricultural practices, you know that. Yes. So nano urea, because urea is soluble in water, if you supply more urea and more irrigation water, urea gets wasted, runoff dissolves in the water and goes away. Plants cannot use it, hence there is a financial burden on the farmer. Yes or no? Nano urea, it will save the quantity and money of the farmer by distributing less amount of urea, but effective nano urea spray. First point. Next is neem coated urea. Neem is what? Very effective biopesticide. Neem. Why? Neem has an alkaloid nimbidin in it. Nimbidin kills bacteria, viruses and fungi, and bitter in taste, repels pests. Hence pesticide, biopesticide, natural pesticide, neem, natural. And if you coat the urea with neem oil, nano neem coated urea, neem oil coated urea, oil coating doesn't make urea to dissolve in the water immediately, then urea dissolves in the water slowly because of oil coating. What is that oil? Neem oil. And neem is a natural biopesticide. Pesticides need not be used if you use neem coated urea. Chemical pesticide usage can be decreased. Less quantity of urea can be used because of nano urea, and urea doesn't dissolve immediately, slowly it dissolves, hence wastage of urea can be reduced using nano neem coated urea. So 3-in-1 advantage. That is one simple application we were looking at here. One aspect, clear? One aspect. You will understand this. You can coat any surface with nano silver and make it antibacterial, antiviral, antifungal. You will understand this. Nano silver. It doesn't cost much. Nano, very thin, very small quantity is required. You will understand this. Yes. Next, titanium dioxide, very popular. What is that here? Sunscreen, lotions and paints, because it can reflect and scatter ultraviolet radiations. So can we coat titanium dioxide to exterior paints of precious structures to prevent fading of or the photo degradation of some marble structures? We can. So titanium dioxide is as of now used in cosmetics to prevent UV impact on the skin. We can apply the same to multiple others. Can we have titanium dioxide nano coating on the car surface to prevent fading of the paint in car when you keep under sun? You can. Now look at applications increase now. Next, fullerene. It is called as Buckminster fullerene. Please understand this. That means it is carbon 60, a circular spherical ball like structure. Carbon allotrope here. Please understand this. So resist pressure deformation. What is resisting pressure deformation here? It maintains structural integrity no matter how much weight you put on it. Nano size carbon experiences or would bear lot of weight. Yes, and gives a sense of fullness in general. So of course side effects are there, but as of now it is used in anti-aging and anti-wrinkle creams to fill the wrinkles. But can we use this to fill the gap if bone is fractured? Yes. Instead of using plate and screw method now if large bones are fractured, it will take a lot of time for them to heal. Hence we put rods inside the bone, or outside the bone we put plate and screw, so that they are brought together and healing happens. Outside the bone we put plate and screw to support two fractured fragments and to help in healing. But instead you can use fullerenes which can support bone from inside without having any side effect. Or in structures made of cement, we can use fullerenes to increase the strength, load bearing capacity. Load bearing capacity is required for bridges, flyovers. You see in India engineers are building bridges which collapse on the second day. Online engineers, yes? Yes, and now flyovers are collapsing, bridges are collapsing, but British constructed bridges are still working. Indian engineer constructed bridges are collapsing within few days or months or years. What is the problem? Materials, quality, and their vision, what they want to achieve, that is secondary. Yes and no. We can use fullerenes in construction also to have the load bearing capacity. Next, carbon nanotubes, previous question here. What is that here? Tear resistant textiles, nano to have great strength and electrical conductivity. In the future, the wires, you see now you can see these electrical wires. If you use carbon nanotubes in the future, you can have invisible wiring, because nano you cannot see it, but it can conduct electricity better than copper. Copper is there, outside plastic is there, rubber, insulation, inside copper is there. Often copper gets heated up, it is thick and costly. Carbon nanotubes, very less expensive, transparent and very thin, invisible wiring. You understand this? That is one application here, yes. And you can also have the cloths which do not tear at all, permanent cloths. Usually many people spend more money on clothing, yes or no? Because as clothes become older and older, they lose color, they lose strength, they lose the integrity, and fibers come out and they get torn off, yes or no? Wear and tear. But what if the clothing never loses color, never gets torn off? Actually you don't have to buy new clothes every time, yes or no? You can wear them permanently. Well, until you are bored of something, yes or no? So tear resistant textiles, stronger and lighter sports equipment, and targeted drug delivery systems. These are few areas where we can have applications here. In general, of course, we can have a lot of such things. We mentioned about them earlier. Yes, that slide we have shown. Okay. Yes. This is a coating on the phones here. That is a tempered glass. Yes, the previous question. Nano electromechanical systems. Yes, flexible variable electronics. Graphene, what is graphene? So graphene is again a carbon allotrope here. So thin and strong, and you can produce or you can develop variable electronics, very thin. Graphene is the answer here, graphene. So flexible electronics or variable electronics here in general, that is one application here. And the nano coating on the surfaces will make them water resistant, and these are applications you have seen them already, where this is what targeted drug delivery by nano electromechanical systems, that is nanobots, targeted drug delivery. It was a previous question, mains question also. So cancer drugs or any other kind of drugs can be delivered during surgery also. To increase the drug mobility inside the body also, they can help, they can be operated. In structural engineering also, we can use nanomaterials which will increase the strength and durability of structures. Camptothecin cancer drug can be delivered directly to cells and cancer treatment here. So those are certain applications of nanotechnology. So what you should know is the working principle behind nanotechnology. Applications are easy to understand, but if you know working principle, everything would be clear to you. Whatever you read in the newspaper regarding nanotechnology, current affairs, you can understand it now better than before. That is the goal. Yes. Next, space technology. We have a lot of such things in space technology here, but still for your understanding purposes here. So what should we understand before we move on to space technology here? So again, space here means what? Outer space. Yes, in general. Why outer space? For two reasons here. Number one is what? For the purpose of what? Observation. And another one is what? Communication. So we have overlapping applications also here. Example, to take pictures, and you know the services, right? Cartography. What is cartography? Map. Yes, and weather monitoring here, and then boundaries, to draw the boundaries, to understand the boundaries here, and eventually to conduct surveys also. Yes, weather monitoring. Disaster management, we know that. Yes, and also natural resources management becomes relatively easy. And in many cities in India, traffic management in the cities is observed through real-time satellite feeds. Yes, so we have a lot of applications here. And then oceans can be monitored, borders can be monitored, surveillance can be given, concerned with larger areas in general. So planning can be one area here, planning of large areas using observation. So observation based services are also called as what? Remote sensing. Remote sensing means observing Earth from space and take pictures and give live feed, so that you can have all these applications in general and compare them across years, so that you can understand change, how things are changing. Yes, and also communications, long distance communications, for example television broadcasting here, and DTH services, entertainment, recreation, and news broadcasting here. And then of course you have something called as the telephone. Earlier we had a telephone, yes, telecommunications, and then now you have mobile. So we are in other parts of the world, I think 6G is going on, yes or no? We have 5G communication. In Japan and other countries, 6G is already getting outdated. Yes, so high speed internet, we mean to say that a high speed internet, we have something called as what? GPRS, Global Pocket Radio Service. Yes, apart from this, when you combine these two, the services will be in the form of what? Navigation, search and rescue. So this helps in live maps, for example if you are traveling, or navigation. So across the globe this would happen here. Yes, so we have a lot of such things here. So how do we do it? In general? Please remember, it is all about what? Space technology is all about designing certain satellites which are human made. Satellites are made with different equipment on board, for various applications, and they are supported with power, battery or solar power, and they should be introduced into specific path. That path is called as orbit, yes, or orbits, and they should be sent using certain vehicle, launch vehicle, and launch vehicles should be designed in such a way that they perform the work, and designated orbit they will introduce this equipment here. And launch vehicles would depend on what? Some physics here, basic. What is that here? Propellant. Propellant means what? Fuel plus oxidizer. We have had questions on this as well. Yes, oxidizer means source of oxygen, that's all, so that fuel would burn, providing required thrust. What is the thrust here? Required thrust. Yes, please. It's called as escape velocity. Thrust is required here, and is Newton's laws of motion, action and equal and opposite reaction here. Yes. So escape velocity of that to overcome the gravitational pull here. Yes. So we have all these things to discuss, very basics, starting from propellants, solid propellants, liquid propellants, and then we have hybrid propellants, ionic propellants, cryogenic propellants. Yes, all these things. Next is launch vehicles. So we have largely 2 to 3 launch vehicles in India. What are they? We have PSLV, and we have GSLV, and GSLV Mark III. We have just a few things here. So next is what? Orbits. We have polar sun synchronous orbit, geostationary orbit, geosynchronous orbit, and geosynchronous transfer orbit for space missions. Yes, we talk about orbits here. And then satellites. Satellites. Usually what will be in use will be only satellites and launch vehicles, and space missions. Space missions, for example, because they will be in use constantly. Yes or no? So for example, some space missions like Gagan, after that we have Antrix. So Chandrayaan 1, older ones, and Chandrayaan 2, and we have Chandrayaan 3. So space missions here, and Mars mission, Mangalyaan, we have space missions here, and then we have yes. Next Aditya L1, India's solar mission. L1 is Lagrange point. It's a previous question here. What is L1 point here? As you can, this is a previous question, hence I included that. So the gravitational forces from both sun and earth gets neutralized at one point. That one point is called as Lagrange point or L1 point, and Aditya L1 is at that place. Neither it is pulled by the sun nor it is pulled by the earth. So both gravitational forces work neutrally. That is when satellite remains stable. You understand this? A lot of precision, a lot of study goes behind it. It's not simple. Yes. So Aditya L1, this was a previous question here. Hence, so and then we have L1 mission, some details here. Yes. And then we have the concept of Gaganyaan mission, manned spacecraft here, Vyomanauts. So we have all these things. Yes. And then we have the Indians in the space. In the recent past, we have these people, because should there be a question, which of the following statements about Indians in the space program Gaganyaan is concerned about ISRO? Names are given. Now can you say, how can you ask names? Can you ask UPSC? They can ask anything they want, whether you know or not, as simple as that. Now just in that case, yes. So we have this, and Axiom 4 mission. On board this only we have Indians on board. So something about Axiom 4 mission here, and then so we found ISRO has found a new launch pad here directly, which can take satellites into the space to avoid a detour, and because of which we had lot of failures earlier. So Kulasekarapattinam will be the new launch pad India is going to build. You understand this? So there is a difference here.
So there will be a question in the exam. What is dog leg maneuver? Dog leg maneuver is associated with which of the following? Dog leg maneuver is associated with which of the following? Dog leg. Dog is associated with dog. Dog leg is associated with dog leg. It is connected with what? Now you can understand. Dog leg maneuver. Dog leg maneuver. It is associated with what? The turn that causes a rocket to deviate from straight flight path around Sri Lanka to avoid the airspace of Sri Lanka. As of now, in Sriharikota, that is called as SHAR. What is SHAR? Sriharikota High Altitude Range, Satish Dhawan Space Center in Andhra Pradesh. There is a launch pad for satellites from India. So that is following dog leg maneuver to avoid airspace of Sri Lanka, which increases complexity of launches and may be one reason for failures. Instead, to follow a straight flight path for launch vehicles to launch satellites into space, India is building a new launch pad in Kulasekarapatinam in Tamil Nadu to avoid dog leg maneuver. A potential question. Multiple questions can be framed based on this. What is dog leg maneuver? What is correct about Kulasekarapatinam? You understand this? So we can have a lot of such things. So just one picture, 2 to 3 questions. That is one aspect. So these are some aspects about space technology we need to talk. Yes. So let's have a look at them in general. So space missions, we have all those recent developments, we have those pictures, don't worry about it. Next we talk about what? Propellant and launch vehicles. In brief, we will talk about them. Propellants and launch vehicles. Propellants, launch vehicles, and then we have orbits, and then we have satellites, and then we have applications. Yes sir, these are basic things. And space missions, as you have seen space missions just now, those will be the contemporary developments. Chandrayaan 3, Gaganyaan, Mangalyaan, Aditya L1, Axiom 4 mission, all those things are missions, space missions, which are in use in the recent past. So all these things you are supposed to know. Okay, let's have a look at them. What is the propellant? So propellant is what? Propellant is the fuel plus oxidizer mixture. So why not only fuel? Because there is no oxygen in the outer space. Simple it is. Oxygen is required for combustion of fuel. Okay. So how much or what type it should be and why it is so different? Please remember, there is a concept called as the specific impulse. Specific impulse. Specific impulse is measured in seconds, which indicates the efficiency, fuel efficiency of rocket propellant. Usually efficiency of fuel on ground based vehicles is measured on the distance travelled per unit volume of fuel, isn't it? Fuel efficiency, some people call it as a mileage. Actually it is not mileage, fuel efficiency. How do you measure fuel efficiency of a scooter, a bike, a car? Distance, 10 kilometers per liter, 20 kilometers per liter, 100 kilometers per liter. Distance per unit volume of fuel. That is what is used to measure the efficiency of fuel in a ground based vehicle. But here we are using a term called as specific impulse to measure the efficiency of a rocket propellant. What is the difference? Basic stuff. Yes, here it is measured in seconds. That means the escape velocity or the speed required for a vehicle to move, escape the gravitational pull of the earth and enter the outer space is very high. So the fuel has to burn continuously to provide same amount of speed. The fuel has to burn continuously to provide same amount of escape velocity continuously. Then what would determine the efficiency? The duration of combustion providing same escape velocity. If a fuel burns for longer period of time providing same escape velocity, that fuel is said to be more efficient and is profitable. Yes or no? But here road condition determines, shape of the vehicle determines, speed of the vehicle determines, load on the vehicle determines, but there everything is kept constant. If everything is kept constant, what would determine the efficiency of the fuel efficiency of the vehicle? Then the duration of combustion of the fuel, hence it is measured in seconds. Based on this, types of propellants are present. Types of propellants. For example, solid propellants. Solid propellants. These solid propellants are made of lot of materials like aluminum powder, yes, and nitrocellulose, yes, and we have hydroxy terminated polybutadiene rubberizer, hydroxyl terminated polybutadiene, HTPB, yes. So these are some of the fuels here, yes, along with the source of oxygen, yes. So this becomes a solid propellant. Solid propellants are, they provide a thrust or escape velocity or specific impulse of approximately 150 seconds to 180 seconds. Specific impulse, please understand this, specific impulse, and they burn continuously, continuous combustion providing escape velocity. So there is no stopping. Once you start the engine, the entire propellant has to burn completely, you cannot stop it, yes. So this solid propellant is used in the first stage of all launches. First stage is called as lift off. So it has to overcome the gravitational pull and lift all the weight and achieve the escape velocity speed sooner and continuously burn until the vehicle escapes the gravitational pull or the atmosphere and further. So after the propellant gets completely combusted, that the propellant chamber will be detached from the body of the launch vehicle. That becomes one stage, yes. So usually all launch vehicles in India launched by Indian Space Research Organization, all launch vehicles use solid propellant in the first stage, and same technology is used in missiles used by DRDO. Missiles use same technology, please understand this, solid propellant, same technology. Missiles, they use solid propellants, continuously they burn until the range is completed. Range means maybe short range, intermediate range, long range missiles are there, yes, that is how it is. Solid propellants are used continuously in general. So we have materials used in solid propellants and their specific impulse and when it is used as first stage. Next is second type of propellant called as liquid propellants. So here we use a fuel called as hydrazine. Hydrazine, we have two types here, that is monomethyl hydrazine and dimethyl hydrazine. So it can be unsymmetrical or symmetrical, doesn't matter to you. So along with either liquid oxygen or the dinitrogen tetroxide. So this is one type here, and another type is liquid hydrogen and liquid oxygen. This can also be possible. This is also possible. Liquid propellants. Advantage with liquid propellants is they have relatively higher specific impulse of ranging from 250 seconds up to 400 seconds. And when hydrazine is used, the mixture becomes hypergolic propellant. Hypergolic means explosive. That is the meaning of hypergolic, explosive propellant. That means it burns fast and it provides necessary thrust in a very short duration of time. Hypergolic, explosive propellants. Hydrazine, high specific impulse. Next, we can have cryogenic propellants. So cryogenic means what? Same liquid propellants only, liquid propellant, but stored under cryogenic conditions. What is cryogenic condition here? Minus 273 degrees Celsius, minus 178 degrees Celsius, ranging between these temperatures. If you store liquid propellants, liquid hydrogen, liquid oxygen, that becomes a cryogenic propellant. So what? Cryogenic propellant, because India recently developed its own Vikas engine, which is nothing but, it is also called as CUS, that is indigenous cryogenic upper stage. Especially in GSLV Mark III, third stage is cryogenic for launching heavy satellites in the geostationary orbit or geosynchronous orbit, 35,786 kilometers away from the surface of the earth. It's very far. INSAT series of satellites are very heavy, communication plus weather. You have to launch them in 35,786 kilometers away. You have to carry lot of fuel, that is propellant, with you for longer distances, and a heavy lift, many thousands of tons will be the weight. So cryogenic engine, cryogenic propellants, they have very high specific impulse. They range from 600 seconds up to 900 seconds. They burn for longer duration and provide necessary thrust of escape velocity. That is why cryogenic propellants are very expensive but very useful for heavy lift, long distance launch vehicles. You will understand this. And cryogenic engine, cryogenic technology, India did not develop until recent past. We requested cryogenic technology from Russia. You will understand this, Russia. But China and America never wanted India to have cryogenic engine technology from Russia. It was international issue, yes, because they thought that using cryogenic technology we will develop long range missiles which can target remote areas of America and China. They had their own apprehensions, because propellants can can be used in many ways, multiple ways. For missiles also we can use propellants, yes or no? For example, solid propellants are used for missiles. It can carry heavier payload to longer distances. Intercontinental missiles enter outer space and re-enter and destroy a target, yes or no? ICBMs. Likewise, if we have cryogenic, we can launch missiles in any place on the earth, yes or no? They feared that India may develop such missiles, hence they never wanted, and also they never wanted India to have achievements in space technology. So they have their own issues. Russia could not give us. It was ready, but at the last moment they refused to give technology. So India took a good amount of time and developed its own. So it is good that nobody helped us because we developed our own. Now cryogenic Vikas engine is indigenous. It is very successful. Okay, anyways. So we have very high specific impulse because of cryogenic, hence cryogenic engine technology is very important. Okay, anyways, don't worry about it. So we have these things. Next is what? Concept of orbit. Orbit is imaginary path. Imaginary path in the outer space, in which the artificial satellite would move around. So movement. So based on the applications and based on the type of satellite, we have multiple types of orbits. So we have polar regions, polar, yes. So one orbit is what? So which is a vertical orbit? Please understand this. I don't know how to put this. Yes, vertical orbit here. Yes, vertical orbit. Is called as polar orbit. Yes, polar. And the poles around a horizontal axis. Horizontal axis. It moves in a horizontal axis, that is vertical movement, pole to pole, polar orbit, and is also called as, this orbit is also called as polar sun synchronous orbit. So this orbit is used by ISRO largely for launching PSLVs, polar satellite launch vehicle, polar, just understand the term, and it is used for remote sensing purposes. To launch remote sensing satellites in polar sun synchronous orbit through PSLV, polar satellite launch vehicle, we use this. And in general the distance is approximately 1200 kilometers to 2000 kilometers in general. And why it is called as sun synchronous? Because anytime you move either from east or west, sunlight is always incident on the earth, part of the earth where this satellite wants to take images. So you can use the sunlight, advantage of sunlight, to take the pictures. Hence it is sun synchronous. You will understand this. Pole to pole sun synchronous orbit. It moves in accordance with the sunlight being incident on the earth. Sun synchronous. We have another orbit called as geosynchronous. Geosynchronous orbit, satellites in that orbit will move in accordance with the movement of the earth, but it is sun synchronous orbit. It moves in accordance with the sunlight. So anytime you want to take picture, sunlight is available, hence picture would be clear. So taking pictures from space is called as remote sensing. Applications are different. Why you take picture is different. You understand this? But we take pictures through cameras on board satellites. We can visualize large area and we can zoom also, but not very precise, from that distance. Yes, that's how it is. So we have these things. First orbit, very popular for remote sensing satellites in India. Another satellite, of course, we have this. Imagine this is the imaginary line equator. Yes, around a vertical line, vertical axis. Yes, so we have horizontal orbit, please understand, in line with the equator, This is at 35,786 kilometers approximately from the surface of the earth. This orbit is geosynchronous orbit in general, and objects in this orbit move in accordance with movement of earth in general. So usually this orbit is used by ISRO for launching communication satellites, long distance communications, television, broadcasting, wireless communications, and satellite TV, that is a DTH, direct to home, and mobile communications and mobile internet. So all these applications are used using INSAT series of satellites. Along with this INSAT series of satellites, we have ocean monitoring, ocean observing, weather monitoring satellites are also launched in this orbit in general. So these are certain popular orbits here, popular orbits in general. And next we have something called as the launch vehicles. Of course, we have two major launch vehicles. If you look at present day launch vehicles, number one is PSLV, which is a 4-stage launch vehicle, and we have GSLV, which is a 3-stage launch vehicle. Please understand this. And PSLV usually is used for launching many satellites, especially the Indian remote sensing, that is IRS system satellites, remote sensing satellite, Indian remote sensing IRS satellites. Whereas GSLV is used for GSAT series of satellites, that is INSAT series of satellites, 3-stage, 2-stage. In both, that is in PSLV, first stage is solid propellant, second stage is liquid, third stage is solid, fourth stage is liquid. Whereas in GSLV, first stage is always solid, because in all launch vehicles first stage is solid, second stage is liquid, third is cryogenic upper stage. And recently India has developed GSLV Mark III, largest, heaviest launch vehicle ever launched by ISRO, GSLV Mark III. Okay, fine. So these are very popular, yes, launch vehicles which are currently in use, and PSLV is considered as the workhorse of ISRO. What is workhorse of ISRO? With a very minimum less than 5% of failure rate, PSLV is considered as one of the most successful satellite launch vehicles in the world. That is why many countries approach India to launch their satellites using our launch vehicle in the designated orbit. And for that we have one office called as what? There is an office called as Antrix Corporation. I think we just mentioned that also for the purpose of it, because we never know. Antrix, this is Antrix Corporation. Antrix is Antariksh. Antrix means what? This is a commercial wing. So directly working under Department of Space and ISRO. So commercial means what? So it links satellites, ISRO satellites, launch vehicles with that of industry and various institutions, research and academic institutions are linked with it, and satellites can be launched. Other countries or ISRO can launch satellites through Antrix Corporation. Launch vehicles can be given on rent, that means other countries' satellites can be launched through our launch vehicles, and various applications, mission support and space sciences are supported through one organization called as Antrix Corporation. It is a coordinating body of many agencies, Antrix Corporation is Antrix, that is what we mean to say here, Antrix. So, because of the trustworthiness of this PSLV with a very less amount of failure rate, many countries approach India, yes, to launch their satellites. And in India also many universities also launch nano satellites, micro satellites. They go and approach Antrix Corporation, and ISRO headquarters is in Bangalore, I think in Penia or somewhere, yes, Penia, third stage or second stage, ISRO. My brother did his project for engineering in ISRO ISTRAC, satellites tracking system is there, ISTRAC, yes. So, getting back here, so these are very popular launch vehicles concerned with ISRO here in general. So, what do we have next? We have all these things, yes, okay. The basics are done here, yes, okay. So, next is applications and missions, that is when this these pictures would help you, yes. So, getting back here, applications, previous year questions, if you look at it, a very popular question is what? India's regional navigation satellite system, also called as IRNSS. So, just for your comparison purposes here, where are we? We are here. We are here. This was a previous question 3 times in the last 10 years. 3 times we had a question on this. Just a comparison. For example, very popular one is what? Very popular. Many people use. Yes. It is called as what? The satellite based augmentation systems for navigation, search and rescue operations, GPS. Yes. So, we know that it is United States thing, and then we have some issues, strategic, security, defense. We cannot trust other countries. Yes. That is why every country developed its own navigation system for strategic, confidential and military purposes. We have to develop our own. Yes. And Russia has developed its own GLONASS, and the European Union Galileo, and China developed BeiDou, and then we have India developed its own, what is that? IRNSS. Previous question multiple times, that is why we are talking about it here. Yes, of course all the details are here, but still, yes. So, 7 satellites constellation here in general. So, it is called as what? Navigation with Indian constellation, Navik also is one previous question here, yes. So, it covers an area of 1500 kilometers radius around India, and then 2013 for the first time it launched here, and each satellite remains for 12 years. So, by 2025 last year, the lifetime of many satellites in IRNSS completed. That is why this time it could be a question. You understand the point here, because 12 years is the lifetime of each satellite. 2013 first one was launched, that means its lifetime is over. That means we can have one focus on it. That is how sometimes questions come. Yes. So, 20 meters for civilian, 10 meters for military is a accuracy in general. Okay. So, what is that we have? So, 7 satellites are there in IRNSS, 4 geostationary and 3 geostationary and 4 geosynchronous satellites are there here in general, and the orbital altitude, that means distance, is approximately 36,000 kilometers from the surface of the earth. Here you can see that lot of equipment are there on this in general, and it will be continuously monitoring the earth here, as you can see that here. Yes. So, 4 satellites are geosynchronous here in general, yes, and then 3 satellites in geostationary orbit, we have 7 in general. So accurate position and navigation, transportation for both civilian purposes and defense purposes. So this is about regional navigation service here. That is one application. This is one application. As we mentioned earlier, other applications are very popular. What are the applications as we mentioned earlier? Number one is remote sensing, as we mentioned earlier. Remote sensing. Another one is INSAT, communication. Communication, broadcasting, internet and positioning services. So these are the general applications, and recent missions, we will have a look at them. Yes. Gagan, what is Gagan here? Have a look at it. GPS aided geo augmented navigation. We have Gagan also here. It is a pretty old system now because it takes the help of GPS. GPS is United States. So now we developed IRNSS completely, but earlier we had GPS help. But GPS did not provide certain services for us, and it started observing our defense establishments also. Hence we developed our own. So that is another story here. Yes. So next thing is what? Antrix Corporation, we had a look at it. And then this old thing, what is that here? So 2008 we had Chandrayaan 1, that is the remote sensing of moon to observe lunar surface. Just we had this. Yes. So it did not complete the mission duration, but it completed mission objectives. We know that. Yes. So we had a lot of other aspects here. You can see that. Anyways, facts are trivial. Nobody is going to ask about on which date satellite was launched. So not all these things. Only the recent ones will be asked about you in general. Chandrayaan 2, again please observe this year. When is that? 2019 September. Yes, that is how we had this Chandrayaan 2 here. And then again orbiter and lander and rover. So three components are here. Orbiter, it will be orbiting around the lunar surface, and then lander, soft landing, which crash landed. We lost communication with it, we know that. Yes. And rover, this also got damaged. Rover means which moves on the surface of the lunar topography. Luna means in Russian language, Luna means moon. Selena, moon. Luna, selena means selenium. Selenium is a type of rock on surface of moon. Selena, selenium. Selena, moon, Luna, moon. So that's how it is. So next is what? Chandrayaan 3. So we have this 2023. Yes. So this is we have one aspect here. What is that here? So we have mission life is 14 Earth days only, short duration here. So and then we wanted to study the presence of water on the moon and other aspects here. So that is what is Chandrayaan 3. We have a lot of equipment. Again, these are trivial, facts are trivial. Don't worry about all those things here. Yes. And then we have the mission planet. So we have red planet here, Mars here again. So we have a lot of details. All the facts are present in front of you. You can have a look at Mars orbiter mission, which is MOM, which is very popular as what? MOM. So these are the details of MOM here, one mission here. And Mars orbiter mission in general is considered as what? The cheapest mission here, very low cost mission concerned with India. So we have NASA's MAVEN, just for comparison purposes here. MAVEN is America's Mars mission, and then we have European that is Mars Express orbiter mission here, and Japan's Nozomi mission here, and then Russian mission and India's Mars orbiter mission or Mangalyaan. So we have this here. Lowest cost. So that is what it is popular as, lowest cost here. I guess it was again we have mentioned here, lowest cost. Among all Mars missions, India's Mars mission is the low cost. That is why it is so popular. In ISRO is so popular because it completes every mission at a very low cost, fractional cost. Like a movie we make in India spending few hundreds of crores, we send satellites across planets. So satellites, so that is so cheap here. So Mars mission, India's Mars mission, as you can see that here, this is United States NASA's Mars mission, and the cost is $671 million, and India's Mars mission is only 69 million. So almost 1/10. What? They are surprised, NASA people are surprised. How can you build and send a satellite at 1/10 of our cost? That means we rely on technology more, not on show off our expenses. We cut down unnecessary expenses. But again, depends. Many Indians go to America to become astronauts in NASA because they pay well. But you know, things are very different. Yes. So Aditya L1, you know that to observe the sun surface. Again, solar remote sensing. See, Chandrayaan is lunar remote sensing. Aditya L1 is solar remote sensing. So we wanted to observe sun surface in general. Yes. So outer corona, magnetic field and solar winds we want to study. So all these aspects here, and hence we have sent the Aditya L1. What is L1 here? As you know that, because that was a previous question here. L1 is a Lagrange point. L1 is a Lagrange point, we say here. Yes. Yes. So L1 Lagrange point here, that means what? Where the gravitational pull of sun and earth neutralize and the satellite doesn't fall and would remain in place in general. That is Aditya L1 here. So this is what Aditya L1 Lagrange point here in general 2023. So that is what is still under operation. So that is one aspect here, L1. And next is Gaganyaan mission, you know that. Again, the approvals have been done, but we are still waiting for this. But the launch got delayed, hence Indian astronauts were sent on another mission, that is Axiom 4 mission. Please understand, Axiom 4 mission is a launch vehicle transferring astronauts into ISS, International Space Station, which is approximately 400 kilometers from the surface of the earth, the International Space Station, to study the outer space environment and to use ISS as a launch pad for extraterrestrial exoplanet or extraterrestrial and the launches, future launches into the space. So that is what International Space Station is all about. And of course, Indians have been sent there. All the astronauts have been sent. This is the total crew of the Axiom 4 mission here. And Indians made their way successfully, and they reached ISS, and of course they came back. Of course we have this here. This is Axiom 4 mission, and this is Kulasekarapattinam's significance here. What is significance? To avoid dogleg maneuver and to avoid any crashes and to avoid moving over Sri Lankan airspace, we are using Kulasekarapattinam, which follows a straight line into the outer space launches. Even if any accident is there, directly it will fall into Indian Ocean, where we can go and collect the debris. So to avoid dogleg maneuver here, yes. So why dogleg maneuver should be avoided? It consumes more fuel, and payload capacity of carrying capacity of the launch vehicle would be compromised, and there is a risk of accidents, and Sri Lankan airspace will be moving into Sri Lankan airspace. To avoid all those things, we go for Kulasekarapattinam, second launch pad in India. So these are some aspects about space technology concerned with you, and recent missions also. That should be enough about these things now. And next we'll move on to defense technology and information and communication technology. Try to go for a lunch break now. It is 1 already, and after the lunch break we try to come back. Clear? Done? So thank you. Go for lunch break, and we'll see you after the break. Thank you.